So, what is your name? There's no mistake. There is a badge. There's no. For the
I think I have some words. So that means yeah. One was left. I love you already. So someone is from here or
no, that's for the editing that would take.
okay. They have that for us. The other thing is that in some of the you know, from after the presentation, you can some questions from people that are so I think there should be some stream.
No, it is a stream, but it's not just a stream. Not participation.
And everything is ready so far? Yeah. Okay.
Do the first
So maybe we can wait for a few more minutes, or it's time.
Do you want to
At the same time. And then we are going to start. So just as a reminder, please, when you say we are not leaving between talks. Yeah, so we are staying here for the whole session. We're going to start in a few more minutes.
This one, yes. So you can press Control L.
Control L. Wait. Full screen.
Full screen, yeah.
So, wait maybe 1 more minute and then we start.
Okay. And for the video thing, is it this one? This one? So,
Let me check the mic.
see my face.
I haven't checked that, so please, if you could check that, you know. Just a second, but we are going to double check that everything is connected for streaming.
Perfect. Thank you. Okay.
So I think it's time to start. So welcome everybody. I hope that you had a lovely lunch. So now we have here for the first session. My name is Gonzalo Chastafilo from the University of Oviedo in Spain. So today we have the honor of having this jury that is composed by, I'm going to read, sorry, my horrible pronunciation, by Pier Paolo Falco, from here from Guerrilla March. Then Stefan Gasparini, yeah, from Sorbonne University, and Cesar Morelos from the University of Oviedo in Spain. So each of you are going to have 15 minutes to present your thesis, plus another 15 minutes with questions from the jury. Please stay here the whole session, don't leave between. And then if we have time, of course, we are going to give you the opportunity to ask some questions, and also, if it's possible, the people that is connected. So whatever you want, the floor is yours. So the first talk is by very neatly, and it's about carbon system dynamics in the Brunswick Strait, Antarctica. during Austral Summer 2019. So, the
Yes.
floor is yours.
Okay, hello everyone. My name is Riley, and as you just heard, I'm going to be talking to you this afternoon about the carbonate system dynamics in the Bransfield Strait, Antarctica, during the austral summer of 2019. So the Southern Ocean covers only about 20% of the total global ocean surface, but it has sequestered around 40% of the total oceanic uptake of anthropogenic carbon dioxide. So this region is very important for the carbon dioxide sequestration of the global ocean systems and makes it a very interesting place to study the carbonate system. So one region, the Antarctic Peninsula, located right here, is undergoing currently a lot of change. It's currently experiencing one of the most rapidly warming regions in the world, significant increases in sea surface temperature, an increase in freshwater input from melting glaciers, and it is predicted to be the next area after the Arctic to experience full calcium carbonate undersaturation. So as you may or may not know, 97% of all of the carbon in the ocean exists in the form of dissolved inorganic carbon, or DIC. So DIC is defined as the sum of carbon dioxide, bicarbonate, carbonate, and carbonic acid dissolved in seawater. And when more carbon dioxide is sequestered in the ocean undergoes processes of ocean acidification, and this decreases the pH and also decreases the amount of available carbonate ions in the ocean water, making it difficult for calcareous building organisms to build and maintain their skeletons and shells. And the ocean's primary buffering capacity against ocean acidification largely relies on its total alkalinity. So this is the sum of bicarbonate, carbonate, borate, and hydroxide ions that are dissolved in the seawater. And together, DIC and total alkalinity make up two really important parts of the carbonate system, and it's what I chose to focus my thesis on. So I won't take you through all of the chemical formulas right now, but it is important to understand the main processes that impact DIC and total alkalinity. So one major process is physical processes. So in this region, importantly, that's freshwater input from melting glaciers and ice. And this has a diluting effect on DIC and TA. So that means that DIC and total alkalinity decrease. Biological processes also impact DIC and total alkalinity. So photosynthesis removes carbon dioxide from the water, which decreases DIC, while respiration increases it. And calcium carbonate also impacts these parameters. So dissolution, where carbonate and bicarbonate ions are dissolved into the seawater, increase TA and DIC, while the formation removes these ions and decreases it. And then lastly, sea air gas flux, where more atmospheric carbon dioxide allows for the oceans to absorb more carbon dioxide, which increases the relative DIC. So one region of the Antarctic Peninsula is the Bransfield Strait. This is an important transition zone between the Bellinghausen Sea in the west and the Weddell Sea in the east, and holds high ecological importance due to the dense krill aggregates that form here. It's also a krill recruiting ground. And as we know, the Southern Ocean food web really relies a lot on krill, so this region holds a high ecological importance, and changes to the carbonate chemistry of this area may have profound effects throughout the entire Southern Ocean food web. So the Bransfield Strait is very variable. As I said, it's a transition zone. So in order to characterize some of the variability of this system, I chose to do both a spatial and a temporal analysis of the carbonate system. So for the spatial analysis, I looked at the contributing water masses and how each of them contributed to the carbonate system of this region. And then I also did a seasonal analysis. So I looked at how DIC changed from winter to summer and the primary processes driving those changes. All of this is with the aim to characterize some of the variability of this region so that we can better understand how the carbonate system might change under future climatic conditions. Data for my study comes from a research cruise that went out in 2019 and collected CTD profiles, which captured the salinity, temperature, and depth of the water column. And they also took water samples at discrete depths for dissolved inorganic carbon, total alkalinity, and nutrient data. So this area is very well understood from a physical oceanographic perspective, and the general circulation is such that relatively warm, fresh water from the Bellinghausen Sea enters the strait from the south and travels along the South Shetland Islands to the northeast. And the Weddell Sea water enters around the tip of the Antarctic Peninsula. It's relatively cold, relatively salty, travels southwest. And then there is a small intrusion of modified circumpolar deep water, which comes from the Antarctic circumpolar current between the South Shetland Islands, and this is identified as a very warm, very salty water mass. So these distinct water masses were easily identifiable on the temperature salinity plot, where we can see the Bellinghausen water as this relatively fresh, relatively warm surface layer, and then the Weddell Sea water, which makes up the majority of the water column, as this colder, denser water mass. And then we can also see this intrusion of modified circumpolar deep water along the South Shetland Islands. So as I mentioned, this analysis is broken into a spatial component and a seasonal component. So for the spatial component, I used the average values of the DIC and total alkalinity values based on the temperature and salinity criteria that I just showed you. And then the pH and saturation states come from the CO2 SIS program chemical model, which requires the input of at least 2 measured parameters from the carbonate system. So in this case, it was DIC and total alkalinity. And then it uses in situ temperature, salinity, and pressure from the CTD cast, as well as the nutrient data from the water samples. And it's able to compute the entire rest of the carbonate system, of which I chose to also focus on the pH and saturation states. And then those are also average for the water masses. And as for the seasonal analysis now, so in oceans that are difficult to sample in the winter time, we can actually approximate the winter surface conditions from the summer sampling. So how this works is that in the winter time, the top layer of the water grows very cold without any sunlight and very saline as the sea ice injects brine into the surface layer. And as the summer sun returns, this top layer of water becomes warm and fresh as the sea ice melts, and it caps off this winter water layer. So we can actually identify the winter water as the temperature minimum shallower than 200 meters depth. And then for the summer water, we took the top 10 meters as the summer water to capture this warm and fresh layer. So then we have the measured summer DIC values and the measured winter DIC values, which we can look at the change. And we can break that down into the 4 contributing processes that I mentioned earlier that change DIC, that cause changes to DIC seasonally. So for the first term, seasonal changes to DIC due to physical mixing inputs such as advection, upwelling, and freshwater are captured in this first term. So this is estimated as the total measured seasonal difference between the summer and winter, subtracted from the salinity normalized difference. And this is salinity normalized to the winter water salinity value. And this is so that we can then isolate the changes in DIC that were purely from the physical mixing processes. The second term captures changes in DIC that were due to biological processes, so primarily photosynthesis and respiration. And we use the nitrate values to approximate this change. We also salinity normalize the nitrate values to the winter water salinity. And then because we know that phytoplankton uptake carbon and nitrogen in fairly predictable proportions, we can then multiply that by the Redfield ratio to estimate the corresponding change in DIC due to just the biological activity. The next term captures changes in DIC that are due to the dissolution and precipitation of calcium carbonate. So this uses the total seasonal difference in total alkalinity from winter to summer, subtracted from the changes in total alkalinity due to the physical mixing processes, subtracted by the changes in total alkalinity due to the biological processes, so that we just have this residual total alkalinity term. And then we can, because calcareous building organisms remove carbonate and calcium from the seawater, it lowers total alkalinity in DIC at a ratio of 2 to 1, so we can then divide that by 2 to get the corresponding change in DIC due to calcium carbonate formation and dissolution. Okay, and the final term, the residual term, captures changes in dissolved inorganic carbon that are otherwise unaccounted for once the changes due to physical processes, biological ones, and the calcium carbonate formation and dissolution have been removed. So this term primarily captures the sea air flux, so CO2 sequestration and release by the oceans, because that's otherwise unaccounted for. And this term does also include all of the uncertainties from the other terms, which I'd be happy to talk more about in the questions. So these were the results from the seasonal analysis, where we can see all of the stations and then the processes contributing to the seasonal change in DIC. We could see that overall there was a significant uptake in DIC from winter to summer, where the physical processes, which are in blue, and the primary production, which is in red, or the biological process in red, showed the largest contribution to the seasonal drawdown of dissolved inorganic carbon. And then in green, we see that the changes due to the calcium carbonate formation and dissolution acted as a sink in some places and a source of DIC in others, but overall contributed relatively insignificantly to the total inorganic carbon budget. And then lastly, we can see this residual term, which showed a very strong positive contribution to the budget, which we can infer is this atmospheric drawdown in the summertime of carbon dioxide. And as for the spatial analysis, so we see that the surface water, the Bellinghausen water, had the highest pH, so it was the most basic of all of the water masses present, likely due to primary production. And then the Weddell Sea water, which makes up the majority of the water column, actually had a relatively high total alkalinity, and that the modified circumpolar deep water, while it is spatially small, actually showed the most corrosive characteristics of the three water masses. So while the values are similar to the Weddell Sea water, it did overall have the lowest pH and the lowest saturation state, so it's the least conducive to calcareous building organisms, and had the highest concentration of DIC as well. So the key takeaways from my presentation that I hope you come away with is that the spatial variability in the Bransfield Strait is largely dependent on the biogeochemical properties of the water masses that converge and mix in this very unique place. The surface waters were characterized by this very warm, very fresh, and relatively basic surface water, which had low DIC as it was likely consumed during the summertime. And the Weddell Sea water, which made up the majority of the water column, actually had relatively high total alkalinity, which suggests that in the future this area may actually show some resilience to ocean acidification. And that the modified circumpolar deep water had the lowest pH and the lowest saturation state. This could mean that this water mass is a future source of corrosion in the Bransfield Strait and would be important to continue to monitor. And then as for the seasonal variability, we saw a significant uptake of DIC from winter to summer, where the physical and biological processes were primarily responsible for the seasonal drawdown of dissolved inorganic carbon. And the calcium carbonate formation and dissolution was a source of DIC in some places and a sink in others, but overall contributed kind of insignificantly to the total inorganic carbon budget. And that the residual term showed a strong positive contribution, so suggesting a strong drawdown of atmospheric carbon dioxide during the austral summer. So just to put this study in a bit of context, the Southern Ocean, we know, has absorbed a lot of the anthropogenic emissions of carbon dioxide, and it's considered to be the next climate change hotspot. This will be an important region to study the carbonate chemistry because it's going to be rapidly changing in the next decades. And the Bransfield Strait holds a specific ecological importance due to its dense krill recruitment, which form the base of the food web in the Southern Ocean. And while this area is very well understood from a physical oceanographic perspective, much less is known about the carbonate system here, and especially how it varies naturally in the system, both spatially and temporally. So the aim of this research was to try to characterize some of the spatial and seasonal variability so that we can better understand what changes under future climatic conditions may mean for the carbonate system in this region.
Okay, thank you very much. Thank you very much for the perfect time. So now we have time for some questions from the jury. Okay, Paul, when you want to start.
Yes, thanks for presenting something about Antarctica. I'm very familiar with this environment. What is not clear, you just used 1 year data, right?
Yeah, just one sampling effort.
How do you expect this time variability can be representative of the global, of the total time variability of the year? Because you just showed 1 year.
Like just one season?
Yeah.
Yeah, I mean, I think it would be great if this data could continue to be captured because it is sampled a lot in the summer. Like, it's a very popular region for physical oceanographers to go to. So if there are scientists able to capture the chemical data as well, we can keep doing the seasonal budget relatively easily. And it would be important to track the changes, maybe not year to year, but over the next 5, 10, 20, 30 years. I think it would be really interesting to see how those processes change under climate change.
Do you think to go on with this observation there, or you just?
Well, I personally,
Yeah.
I would love to. Yeah, I had the best time doing this thesis, so that would be great.
Which is the size, I would say, of the area you explored? Because you speak also about spatial variability. So basically, based on, I guess, to the water mass you observed there, right? So
The what,
the
sorry?
spatial variability.
Yeah.
So you study the spatial variability in terms of water mass distribution.
Yes.
Which is the dimension of the area.
I can go back.
The size
Do you mean
of
like the sampling?
Yes, because you didn't see any.
Yeah, these were the sites that were sampled. So it's not, there could be better spatial coverage, which was, definitely a limitation of my study, but that's the situation. I think that they ran into like an iceberg around here also, was the story I was told, so they had other stations planned down here, but there are also a lot of stations that are kind of like known repeat stations that they just go back to every time they're in the area.
Very good presentation.
Thank you.
So now, yes.
Yeah, congratulations, man. It's almost done. I'm a little bit surprised that are these the first data in this area? Because, you are presenting a characterization, but can we compare this with something? There are previous data, there are data in other areas that can give you some kind of signal. What was exactly your expectation when you or your tutor or the supervisor in the research group is doing this? This is important, no?
Yeah, I understand your question. They have done, there's not a lot of carbonate system data in this area, so I did find some similar spatial analyses done where they were trying to look at how the water masses contributed, but in terms of like the temporal variability, I didn't find anything in the North Antarctic Peninsula. I did find one study done down more south along like the western Antarctic Peninsula down here that did this same seasonal analysis, and they had very different results than I did. Like the contribution of the processes was similar, but they had more clear spatial patterns that emerged for them. They had really clear onshore, offshore gradients in the processes themselves, whereas like I actually didn't see any patterns in the processes, which I think is largely because it's a semi-enclosed area, so it doesn't have the same onshore, offshore characteristics. So yeah, to my knowledge, this was the first like seasonal, temporal study of the carbonate system in the North Antarctic Peninsula.
In that area, but
Yeah.
you said that this will be possibly the next hot spot.
Oh yeah.
For climate change, but there are other areas where something has already been changing. So
Yes.
it's like the prediction or the initial hypothesis about, okay, how is the liver now? How are what the factors contributing to this, and what we can expect them? Because in other areas we have observed this and this and this, and if this will be the next hotspot, probably this will happen, and what we can do, because at the end is...
So what can we do? Is that the question?
No. The question is, if in other areas there have been already data this type with changes, and that maybe can help you to predict what will happen.
Okay. That I do not know. I only was reading about this one study I found on the Western Antarctic Peninsula that did this similar seasonal budget technique. Yeah.
Okay.
Yeah. Yes. Thank you.
Thank you for your presentation. It was a very nice piece of descriptive oceanography. As you know, I'm a biologist.
Yep.
I'm not a chemist. I'm not well detailed about the carbonate system. So I will ask you 2 questions about biology.
Oh no. My nightmare.
My first question is about your estimate of variation in inorganic carbon due to biology.
This one?
Yes.
Yeah.
This one. You explained that you multiplied nitrogen concentration by the red field stoichiometric ratio.
Yeah.
Could you please explain which one of the red field ratio you use? Because there is many ratios actually in the original paper of red field.
Okay.
And probably you use the one concerning one part of biology. Which one?
I used this one.
That's this one. So this one I recognize it. So
Yeah.
this correspond to what part of the biological?
What does it mean? Or what is it?
I mean in the plankton. This ratio corresponds only on one part of the plankton. Which one?
It is the.
The answer is very easy.
Oh, okay.
Don't search for very complex.
I feel like I don't totally understand the question, but it is the uptake ratio of carbon to nitrogen by phytoplankton. Oh, okay. Yeah.
The answer is it is limited to phytoplankton.
Phytoplankton, then? Yes.
So it's not a
the
very
answer.
difficult question.
Okay.
So, but it's important because sometimes phytoplankton is not dominating the system in some circumstances.
Yes.
My second question, so it will be very short. It is about your final conclusion.
Okay. This one? Or?
The final conclusion, yes. The second line, okay, you are talking about the importance of this area due to very dense krill recruitment.
Yeah.
I agree with that. But
Okay.
do you think that change in the carbonate system could affect the krill, and why?
Yeah, so I wrote about this a bit in my thesis, but they're already seeing changes to the planktonic community, the phytoplankton community in the North Antarctic Peninsula, and especially they're seeing a shift from larger cells to smaller celled organisms, just with the changing conditions of the water. And krill have a very specific grazing capacity or grazing range, and when the organisms that they graze on get too small, they get clogged and they actually can't.
So you think that change in pH could change the size of phytoplankton in the area?
I think it's
What's the main component of the phytoplankton in the area? Do you remember?
the main component.
The main component of phytoplankton in this area.
The main type of
Phytoplankton, it's
diatoms.
mainly mainly diatoms.
Yeah.
And are diatoms sensitive to pH?
They have seen a decrease in diatoms in this region. I don't know if it's not pH driven, but they have seen a decline in diatoms, and they're inferring that that's why they're seeing less krill in this area, because that's a primary prey of krill.
Could it interact with the silicate system, or it could be due to another process?
I'm not sure.
It's an open question.
Maybe we'll have to think about that one. The silicate system. Yeah. They have already seen changes though to the planktonic community, and they are linking the size of the phytoplankton to the krill decline, which they are. I think it's also physical oceanographic related. I also had a component of that in my thesis that I just didn't have time for, but looking at mixed layer depths and how they tend to be becoming more shallow in the short term with climate change, and that being less conducive to certain phytoplankton communities like diatoms. They get too much solar radiation, I guess, and don't do as well.
So it's an indirect impact.
Yeah, part of maybe the broader oceanographic implications of climate change, physical side.
Thank you very much.
Yeah.
So I think we have time for one more question from the audience. Yeah.
Thank you for that excellent presentation. I really liked it, and you also made it accessible to people that are not on this or subject or discipline.
Yeah.
I have one question about temporal variability.
Yeah.
What happened to station 6?
Yeah, that is a really good question. That's a great question. My best guess or my best understanding here at this station was that because this was my shallowest station, it was only about 120 meters, and I identified the winter water layer at around 105 meters, which is quite close to the seafloor. And my best guess is that there was other interactions going on here beyond the primary processes that I captured in this seasonal change. And so, yeah, if there was a seafloor interaction with the winter water layer, that could be increased carbon dioxide from respiration from organisms on the seafloor. It could be increased calcium carbonate dissolution due to dissolving skeletons and shells on the seafloor. But yeah, I think it is because the winter water was identified very close to the seafloor.
Okay, that's great. Thank you very much.
Okay, thanks.
So can you pronounce me? So I will let you know, I'm going to be seat when I stand up, you have 3 minutes, and then I'm going to do it like that when you have 1 minute. Yes? Okay? So, can you please? So, then, Laura Champagne is presenting the talk with the title Exploring the Diversity and Biotechnological Potential of Prokaryotic Community Associated with Marine Sponges Species in the Arctic area. Whatever you want, the floor is yours.
Hi everyone, I'm Eloha, and I'm gonna talk about bacteria from the Arctic and their potential in biotechnology. So you probably know the Arctic. It's also called the North Pole of our planet, and it's characterized as an extreme environment, both for humans but also for microbes. Because of temperature below 0 degrees, ice coverage, adverse solar radiation, low biochemical activities, etc. Plus also anthropogenic pressures such as pollution by microplastic and hydrocarbons. So all of that makes it a very hard environment to survive, but it has been estimated that both for Antarctica and Arctic represent 14% of the total biosphere. So how does life survive there? A few years ago, cold adapted bacteria were discovered by Morita in 1975. So it's bacteria indigenous from these cold places that have developed adaptation to survive and even thrive in such environment. So for instance, adaptation can be, they modify their lipid composition so the membrane is more flexible, or they can also produce a specialized molecule or proteins that allow them to resist. And it has been found that some of them also adapt to pollution phenomena, especially hydrocarbons, and can survive it. And even more, it has been found that these bacteria were also discovered within marine vertebrates that cannot survive on their own. For example, sponges, they are sessile organisms, so they're very sensitive to the environment, to what's happening around them. So forming kind of this symbiosis with bacteria that are adapted can help them themselves to survive. So this is the goal of this thesis, because this symbiosis has been a little studied in Antarctica, but not yet in Arctic. So my thesis was to explore the microbiome associated with two marine sponges from the Arctic, Aliclona and Mixilla species, to study the taxonomy structure of their communities and if they have a biotechnological potential. So for this, I did two parts in my thesis. The first part will be metabarcoding to really see the complete diversity of microbial diversity within the two sponges, Mixilla and Aliclona. And the second part is focusing on the cultivable part of one sponge, which is Mixilla. And I tested over different contaminants what they can survive and how they can grow with these contaminants. And my thesis project is part of a bigger project which is called the Symbiosis Project and funded by us in 2020. So to look at this a bit more, the sponges were collected in the Arctic, so in the Svalbard area, in the Longyearbyen area, which is the northernmost inhabited city, inhabited place in our planet. And the sponges were collected at 8 to 16 meters. They collected 3 Mixilla, 6 Aliclona, plus water samples and sediment samples. So, to say it again, I did two parts in my thesis. The first part is metabarcoding on the gene 16S RNA to study the complete diversity of microbes within the two sponges. So we also use Illumina sequencing, and then we analyze the results. And for the second part, I focused on Mixilla. Using internal tissues we cultivated, we tried to isolate the bacteria we could. Obviously, a lot of bacteria are unculturable still nowadays, so we could not isolate all of them. And then we isolated them in selective media, 3 different selective media, and according to also their morphology, colors, characteristics, we made them in different media. And we secret sequencing to know what they are, to identify them. And then we did different tests on them. So I will not have time to talk about all the tests I did. It's all written in my report. I'm going to focus on the tolerance test today. Also, other data that I had, in collaboration with another institute, British Academy of Science, they studied the internal tissues of Mixilla to know what contaminants were present within the tissues. So, first part, to know the complete diversity of microbes within both sponges. So, in the left, it's the communities associated with Mixilla. Here, the communities associated with Aliclona and sediment and water. It's representing the relative abundance of all families and phylums that are superior to 1%. So if a bacteria is present but its abundance is inferior to 1%, it's not shown here. What the result shows is, first of all, for sediments and waters that are the environmental samples, it's much more diverse. There's a lot more families present, more than 1%, of course. This is now, it already have been shown. And for the sponges, we can see that there is, for the two communities, there's different communities, different major bacteria. So even though at the phylum level, Bacteriodota and Proteobacteria are the main one, when we focus on the family level, for Hemicilla, it's Rhizobiacea that is dominant, and for Aniclona, it's mostly PS1 clade and EC94. So even though they're living in the same environment, we can see different communities. To go even further, we decided to do a network plot where, again, still superior to 1%, the strains are present for each environment or sponges. And we can see that they are very well separated. So that's like what is here, Aniclona is the sediment. So that shows that all of these environments host different communities from each other. In the second part of the study, so we took tissues from Klebsiella and we isolated the bacteria, right? We isolated in total 27 isolates that are part of 5 main families. So after single sequencing, we could identify them. So Mycobacterium, Cellulose microbiome, Planomicrobiome, Planococcus, Spirobiome, and one that, despite a very good sequence, has no close relative on gene banks. So the closest relative neighbor was 76%, which is not enough to say what it was. So maybe it was uncultured before or not reported yet. So this is a +1. And all the bacteria, all the isolates that we made grow on selective media under hydrocarbon fluoride were Spangobiome. Also, it's not figured here, but none of them grow on mercury media. So mercury supplemented media, no growth. Now I'm going to talk about the tolerance tests. So we tested some of these isolates and we tested them in a new way that we found on a multi-6 plate, so we can test them according with different contaminants. So we selected 16 different chemicals that we had in the lab and were also found in the tissues during the chemical analysis by the Polish Academy. And we analyzed if we have growth or not. So the result takes this form. It's the growth, bacterial growth is measured using optical density. So if there is an increase in optical density, we say that there is an increase in bacteria, so bacterial growth. Yes, and we choose some of the isolates. So we can see that this is the control, so they grow without under no contamination, and then under each different contaminant that we classified by categories. So heavy metal, hydrocarbons, PAH, hydrocarbons, antibiotics, drugs, pesticide, plastic, and PCB. In the control, we can see that the strains belonging to cellulose microbiome and Planococcus follow quite a similar protein, while the other one, Spirobiome, grow much less even in the control. And in overall, what we found is these 3 main strains, so isolates, cellulose microbiome and Planococcus, show a very wide tolerance to a lot of contaminants, but not all of them. For instance, mercury and gentamicin, there is almost no cross for all of them without distinction, which for mercury is reassuring because we did not see any growth on our media, so it makes sense that we cannot reproduce. So what can we say about all of this? First, in part 1, we found the major family of bacteria present within the 2 sponges. So for Aliclona, it's EC94 and PS1 plate, and for Mixilla, it's Physiobiocide. So they show different communities, even though they were living in the same environment and they were collected in the same place. And they also show different communities that found in water and sediment. So that can reassuring about the hypothesis we made that hosts can select, shape its communities according to what they need, but more studies are needed, and especially evolutionary studies are needed to understand the effects that environment has to push the symbiont to actually choose its own communities. And for the part 2, tolerance profile, we isolated 5 main strains of bacteria, plus one that has not been reported before or has not been published yet. And some of the strains show tolerance to heavy metal, hydrocarbons, pharmaceuticals, pesticides. And they show an ability to survive in polluted environments, so they have potential for future biotechnological application, especially in bioremediation. But even though if this result can be encouraging for bioprospecting them, it is just an exploration inside these symbiont communities, in the communities of these arctic sponges, and a lot more studies are necessary, especially to culture more of this bacteria, because the bacteria we could isolate were present in less than 1% in the total communities. So I think it would be a good work for the future to improve these culture abilities. Thank you.
Yes, these topics are very far from my background, so it was hard for me to follow your presentation. Anyway, just curiosity about the contaminants. Do you think that there is indication of the human impact from them or not?
I think so, because Arctic is starting to be more polluted, and because of the cold environment, the pollutants stay a longer time. And for example, for hydrocarbons, due to the boat trafficking or things like that, there is a lot more hydrocarbons. And studies were made that PAH were even found in some rivers in Arctic. So I think so. Also, when we did, when we did the chemical analysis, we found inside the tissues, we found contaminants. Especially pH, there is quite high level of pH in them. So.
And you can discriminate between local contributors and
natural ones.
Yeah.
There is local contributors and natural contributors because of the hydrogen bands and things like that. Yeah, there is contaminants from. Not only from humans, also from natural reasons.
Because in the past there was some discussion about the impact, for instance, in your sea of the waste. So I was wondering just if there is some indication that the local human activity on the island can impact.
No, yeah.
Thank you. Very good.
Thank you.
Okay, congrats. That's the first thing. And so the initial hypothesis is, okay, living there is very difficult, so those organisms could have some help, and maybe bacteria can play a role. And then you are finding that maybe it's not a global pattern, because each species of sponges, they have their own community.
Yes.
But you find something, because you put in your network, you put some bacteria that were shared.
We shared, yes. When you look at it,
So,
only one is shared, because the other one is under 9, yes. So, those lactobacteria are shared by all of them, all the samples, water and sediment, which makes sense because they also have water and plants.
Yeah, but it's because, I mean, it would be great if you are able to identify some kind of bacteria that can help those organisms to live in these extreme conditions.
Yes, that's the next step of the
But
study.
not for now.
It's the next step of the study. Also, the bacteria that we found, so the ones that are the main ones, are all unculturable. So it makes it hard to study them. So we need to do metagenomic on them to understand how they can be expressed, what they express, etc. But they're unculturable yet, so it's hard for us to know how do they impact, how do they help.
Yeah, how is the interaction between the bacteria and the... Are there any other studies in other type of organisms in which these hypotheses can be
More
tested?
developed?
Yes?
It's very new, but it's starting with Antarctica. Sponges, I think there were other organisms too, but mostly in Antarctica, who can be quite different from what we have here. And again, they need to do all of them there at the same time, more or less. They did metabarcoding and trying to identify which one and how can they survive, under which contaminant can they grow. But more studies are needed, and especially evolutionary studies, I think.
And are there evidences about an increase in antibiotics? Because you put plastics, okay, everyone knows that plastics are increasing everywhere, but you put antibiotic and you put
Antibiotics?
yes, several other molecules. Are there evidences? Yes. And drugs.
Drugs, yes.
Are there evidences about an increase?
Increasing in drugs? Yes, I think there is a recent study about antidepressant in Arctic.
Yeah.
So that's what we wanted to test, but we did not have antidepressant with us, so we used paracetamol and ibuprofen. But it's also molecules that can last very long in the cold. That's why I wanted to test them. But it's also to think about future in biotechnology, in maybe industrial, before in bioremediation. So my goal was also to know if they can grow in such contaminated places, if they can degrade it, and if we can use that for bioremediation purposes.
Or something like that.
Yeah.
Good. Thank you so much.
Thank you very much.
Mike, thank you for your presentation first. And my first question is not really a question. Did you go to Longyearbyen?
I wish. No, but I plan to.
So, because I went there a couple of times, so I know the place very well. And,
It's wonderful.
and the community. And it's important, my question, because, okay, how did you get the data? Because my first question is whether the incubation or the experiment done in Longyearbyen, in the Marine Lab, no, in Nias-le-Sud, in the Marine Lab in Nias-le-Sud, or back to
Sicily.
To somewhere else?
Sicily. So I was based in Sicily, in Messina. I did not go to collect samples. It was quite far. It was collected a few years ago by my supervisor. For the sponge tissues.
So it means that all the samples were preserved?
So the tissues were frozen, but they also put in glycerol, so do not tissues that I could use, and yeah, they use the frozen tissues to reduce the experiment.
Okay. So all your results comes from frozen samples.
Yes.
And it leads to my second question. My second question is how you isolate bacteria, again it's a methodological question, from the sponge. Because you are talking about symbionts, but my question is how you separate symbionts from external bacteria, internal bacteria in the sponge, things like that.
So what I did is I opened my sponge, I took the internal tissues of it, I diluted it in seawater, I smashed it very slowly. We tried 2 protocols because the first one didn't work. I think we were too violent, and they were all samples, so we tried another one. So we just took the samples, put it in seawater, smashed it a little bit, did different dilutions, and putting the dilution on marine agar plates, different dilutions, and actually I got bacteria in the pot.
Now we can be sure that it's not contaminated with bacteria from surrounding.
From outside, it's because I took, the sponge was closed. I really took internal tissues for this purpose.
Okay. So I have many questions. My second question corresponds to the figure of your first part.
The network block?
This one.
Yes.
Very nice figure, but I don't really understand the method behind the figure. Is it a PCA? Is it MDS?
It's Illumina too. It's the same data set that I used for this one.
Okay.
I just wanted to represent it differently. So I did, if the strain is present more than 1%, there is a dot for each. And it's exclusive. The colors are, for example, the blue one are only found in water. These ones are only found in Arizona, Ixila,
Yes, I understand that part.
This one is shared between sediment and water, and this one are found in all environments.
My question was about the multivariate statistics behind the graph.
The statistics behind that?
Yes.
That's written in my reports. We did I can show you.
It's a non-metric MBS.
Yes, because I use the same data as I did for the histogram, so we have the same statistics.
Okay. Non-metric MBS using regulatory similarity. It's so it is the answer for my question. Okay, again. I have time?
Yeah, wait. Sorry.
Okay. Do you have any representation about the difference between the culturable bacteria and the bacteria present for the part 1? You have many bacteria.
In part 1,
in
yes.
part 1, a very small number. Second part, of course, it's not a surprise. But what about the difference? Because it's 1%, 10%. You calculate how many, what the percentage of bacteria you retrieve are culturable compared to the total number of bacteria in the first part.
I can give you the numbers. So
I imagine it's a small part, but it was not possible to calculate it.
superior to 1%, because I focused my method barcoding on superior than 1% because I had a lot of different ones, so I removed everything that was inferior to 1%. So superior to 1%, but it's not very comparable then, because the bacteria isolated are present in less than 1%.
Okay.
So that's why they don't appear in part 1 because they were probably a tiny bit within the tissues. that's what I say, that we need to improve controllable methods because we are missing a lot of information because of this problem. And that's the next step.
Okay, so the direct response to my question was less than 1%.
Yes.
Okay. And last question. But it's for my own information.
Okay.
You said you test crude oil.
Crude oil, yes.
And I tried to test crude oil in other organisms, but it is very difficult because it's hydrophobic, of course. So how you solve this problem? Because you said you test it on bacterial strain. So it is in water suspension or you use another solution?
That was a big problem.
I know it's a big problem because I tried.
When I tried at first, so when I isolated my bacteria on this, we use a technique that my supervisor knows. It's on a filter, on a sterile filter. We put drops of crude oil and then we invert the dish. So they're not in contact with the crude oil, it's more the vapor of it, because it's closed. When I tested it in the tolerance test, so here, we decided to not reuse it when we reproduce the experiment for future duplication, because the problem we had, I did a mother solution with crude oil and I think it's DMS. And it mixed very well, it was great. And when I put in the plate, it was well mixed. I made sure for the concentrations that it was well mixed. There was just one problem, was that it wasn't transparent. So the DO calculation was tricky, and that's why we have negative values sometimes. And we cannot really say anything about it. So when we reproduced this experiment for further purification, we decided to remove the crude oil.
Okay.
Till we find a
Thank
way.
you very much, but I understand you did the experiment yourself, and it's a very good job.
Thank you very much. So last but not least, now Kelly, sorry, my pronunciation again,
McLaughlin.
Sorry, it's presented her thesis entitled "The Rainbow Connection: Implication of Changing Shoreline Colorscapes on Marine Biodiversity." Again, I wanted to know, so I'm going to stand up when you have 3 minutes left, and then I will do like this when you have 1 minute.
Wonderful.
The floor is yours, whatever you want.
All right. Hello, everybody. My name is Kelly McLaughlin, and this is the Rainbow Connection: Implications of a Changing Shoreline Colorscape for Marine Biodiversity. So, we begin with a problem. As we all know, global populations are increasing everywhere, and with that, there's also an influx of people moving towards our coastlines. What that means is that there's more infrastructure to accommodate this increase, and it leads to a lot more development along our coasts and creates an increase in shoreline hardening, such as the creation of seawalls, revetments, or jetties to protect this infrastructure from erosion, sea level rise, or storm events. In Sydney, Australia, where this study is based, there is typically a lot of natural ecosystems such as our sandy benthos, macroalgal benthos, and rocky shorelines. However, when we put in and install these, albeit necessary, marine infrastructures, we are taking away those natural habitats and decreasing what is typically there. And when we do this, although it still supports a certain level of diversity, it is contributing to a loss of overall biodiversity, poorer water quality, more reduced resilience, increase in invasive species and non-native species, and as we start to understand and realize that this isn't really something we can change, and we scientists, researchers, and engineers have all begun to ask, instead of how can we decrease our marine infrastructure, instead, how can we make it better, which leads us towards our solution. And that's marine eco-engineering. So all of those folks coming together to figure out how we can create a better, more nature-based solution while maintaining all of these structures. Because we have to live with them, and ecosystem restoration is not always something that can be done. We have to figure out how we can accommodate these marine structures to better support higher amounts of diversity and maintain the natural ecosystems that maybe used to be there. So, how this has typically been approached is by exploring habitat complexity, which shouldn't be so unfamiliar. There are different ways it has been done with different sorts of crevices and structures along seawalls and gradients along different tide lines. But there have also been other considerations as people explore eco-engineering deeper, such as the types of materials that we use, either metals, concrete, rocks, polymers, and the microtextures of those things. But there has been a large gap in the color. And why should there be? Our natural ecosystems are full of color. As you can see here in this wonderful graph made by Holly Cunningham, a former student at Macquarie University, there are large colorscapes along our coastlines as we have diverse spectral reflectance of sandstone, mangroves, and oyster reefs all around Sydney Harbor, which is a stark contrast to the monochrome gray scale of the typical construction concrete that is used in marine infrastructure. But color is really important. It's in our natural ecosystems and shouldn't be neglected when we're thinking about constructing marine infrastructure. Preliminary studies on color have found that larval settlement use photoreceptor-mediated processes to understand what color is, as different organisms have different ways to perceive color. And they've shown that there's also been studies that red substrates were associated with distinct biofouling communities, and further suggests that color can play a role in structuring early stage communities. Also, as larvae settle and they want to biomimic mimic their background, they might choose different colors to better protect them against predation or other factors. But this just leads us to more questions. What can we ask? So, since we already know a little bit about complexity, we can ask about how color and complexity may interact. Will they have a compounding effect or not? Will the effect of color be consistent across different environmental conditions? And will the effect of color also be different or maintained around different microhabitats, i.e. crevices or ridges, biogenic or even larger areas? And for this, the purposes of this study, we hypothesize that yes, both color and complexity will influence colonization, that color effects will vary with habitat complexity and microhabitat. Ideally, the strongest effects of color will be seen along flat areas and ridges with more sun exposure and more direct perception of the color, and that environmental context, particularly better water visibility, will likely play an enhanced effect on color and complexity in areas that are less degraded and less polluted. So, we begin our study. We began by picking 4 study sites around Botany Bay, which is just south of Sydney Harbor. We picked these sites based off of a 2022 paper by Birch, Guns, and Lowndes regarding the different water qualities and metal pollutants from the Georges and Cooks River into the bay, so that we could have a wide variety of water quality and different environmental conditions. These places also did have different other parameters, such as the angle at which the seawall was, the orientation towards the sun, and what benthos were adjacent to the structures. And once we had all these sites, we made all of our panels. So for the purposes of this study, we made these 25 by 25 panels with the help of Reef Lab, and they were across 4 colors: gray to mimic as our control for marine concrete, red, green, and yellow, all made with specific wavelengths to match the natural substrates, as well as all maintaining the same brightness so we could really isolate color's effect. We had 2 complexities, so complex panels with ridges that were at least 5 centimeters deep, and then flat panels with an equivalent planar area, and 5 replicates of each of those 8 treatments. That's 40 panels per site, Across 4 sites, 160 panels to census. Woo! We put them up on the median tidal line and then waited for things to grow. Once things were there, we did biological censuses. Using quadrats, we counted the sessile species at each intersecting point on a quadrat to get area coverage. And so we had 25 points in total, and we did a similar process within our microhabitats. So on the flat panel, that meant choosing a random point to put our micro-quadrat on, as well as for the complex, putting the micro-quadrat either inside the crevice or on top of the ridge, and doing that same intersecting section. And anything else that was on the panel, but not falling under an intersection, was denoted a lower value to just have noted its presence for the total richness. Mobile species were also counted as individuals to have their abundances, and environmental data was also collected using a probe and Secchi-Dis so that we could possibly isolate what environmental parameters may have been different site to site to see if anything was consistent in showing different results. What did we do with all this data? Well, we did univariate analysis for alpha biodiversity matrices, species richness, abundance, and Shannon index using generalized linear mixed models to understand any relationships between our factors and visualize those with violin plots, which you will see later. And then we ran multivariate analysis across all 4 of our sites using Permanova with post-hoc to once again identify what relationships were driving any large differences and visualize those with non-metric multidimensional scaling gloves. And what did we find? A lot of things, but some of our most important takeaways were that complexity did have consistently high levels of sessile abundance at every site. So for this, for my thesis, data was collected at 2 months and 4 months. And so at 2 months, every site had this difference. Here we have island graphs for complexity and flat with the associated colors for every single site. And we can see that at 3 of the sites, complexity had higher percentage cover of sessile species than as compared to our flat panels, while over here at Ramscape, we see a consistent amount of percentage cover. That's just due to its environmental conditions. There was a lot of growth regardless of complexity at 4 months for Ramscape. So we believe with more time, that pattern will continue. In regards to color, we found that color, this is the same graphs, often but not always had an effect. And when it did have an effect, it was different site per site. So it starts to paint the picture that every site is specific and the environmental conditions around each site were factoring into what colors effects were. So you can see here that even within Cars Park, flat panels had a distinct relationship between gray and red, while the complex had large significant differences between red and green. And that sort of pattern continues on throughout the rest of the analysis. Within microhabitat, we found that microhabitat does have an effect at all sites. Similarly, these graphs show all 4 sites, but now isolating each microhabitat. And just as before, 3 of our sites had the significance while Ramscape was more consistent in the abundances at the microhabitat level. And there was also a color effect between the microhabitats. At Kaima, it was within our ridges, and at San Souci, it was on the flat. So obviously we have the greatest differences where color is, as we hypothesized, better perceived on the flat and ridges. However, what was driving the significant differences between the microhabitats was the relationship between the crevices and the flat areas. When we look at the data across all of our sites, the large NMDS plot for all 4 sites together is a bit confusing, so I isolated them for you guys. However, as you can see, the distances between the 2 points are most clustered here at Ramscape, showing that the panels and supporting that had a consistent level of similarity in terms of species richness and abundance, while a site, say, at Kaina has a large disparity between our fill and complex and color, as our reds are all quite similar, while the yellows have a stark difference and are more spread out, similar to the greens. and that further supports some of the color results that we had seen as well. And all of this just basically helps to indicate the importance of the environmental parameters when we want to incorporate color into eco-engineering projects. So what can we draw from all of this? Well, the most important thing that we're learning and that we found was that yes, color and complexity and habitat complexity did influence colonization within these first 0 to 4 months. And there was a main effect across all 4 sites when we just look at color. So disregarding complexity, across all 4 sites, there was an effect between green and gray, red and yellow, and red and gray. All of these ones having the higher abundances and species richness. And across all of these sites as well, the species that was driving and dominating most of these relationships and the significant differences between color and complexity was Ostromenius cuvertus, a species of barnacle found in New South Wales. And it wasn't just the presence of this barnacle, but more so the abundance of this barnacle. So any panel that had higher abundances of Ostromenius cuvertus was showing a large difference between any panels that had lower abundances. What this could mean is just that these species were really driving it, and it also goes to show that at 4 months, when only 22 species total found, we probably need to reach higher levels of total diversity to have more of a full understanding of the effect that color is having. Which just leads us to ask more questions and figure out what other research needs to be done. So, what we need to do and what is being done with this particular project is a longer-term study. Obviously, this is only 4 months of data, so we are doing a project throughout the entire year. 6-month data was just collected, and 12-month data will be collected in December to have a full insight across an entire calendar year. And we want to also continue doing censuses at 18 and 24 months to understand ecological succession or any following trophic relationships that might be influenced by color or not. And we overall just believe that this research has a large ecological value and further supports the insertion incorporating complexity into marine structures, hopefully at a larger scale, as well as the importance of factoring in color. Color is a really wonderful part of our natural habitats and is very specific to each environment. So we suggest that more research needs to be done on area per area of the best combinations of habitat complexity and color specific to a site possibly. And we also think that having larger scales of research would be beneficial to understand, since this is such a small area that we are looking at, if there will be any greater effects or significance across larger scales of complexity being added, such as making an entire wall one of complexity rather than just having small spaces. Overall, it just further confirms that color is an essential part of our ecosystems that shouldn't be ignored and should be further considered when we are talking about eco-engineering and safeguarding our coastal communities. Thank you so much.
So now it's time for the jury to do some questions from the board.
Very good presentation and very interesting work. It's very, very far from my knowledge. So my question is just, did you compare if there are your study with other studies carried out in other regions with different, I don't know, daylight condition, exposure, or whatever?
Yeah. That is really interesting and should be more research. Right now, a lot of this color research that is being done has either been done in situ, so like within a lab, and most of the ones done in the environment that we know of have been from the lab that I was working in. So it's all the same environmental areas and conditions around Sydney. So I would be interested both in other areas doing this research for that same thing of different environmental parameters, but also the different communities that could settle on there. I think that it's limited right now to the several species that dominate in this area, which is also a highly polluted area. So historically, we're not even getting as many species as I think we could. But yeah, and this project is in collaboration too with larger goal of living seawalls. So they do have partners, so hopefully it can take off in other parts of the world.
And about you say that you analyze the short-term series, not just
Yes.
So do you expect that there will be changes in your results if which kind of results can change?
Yeah. For sure. Right before I came here, I did collect the 6-month data, and even within sites, the abundances of certain organisms changed. There was less algae, and it's changing every few months now, which is really exciting. But there's also just not a lot of growth at most of the sites. The 3 sites that have like the most differences just still don't have a lot of growth on them. There's a lot of bare spots on the panels, and this one with a lot of consistent kind of shows a little bit of projection of the future results, I think, because these panels are pretty heavily covered. Almost, you can see 100% and a little bit more coverage. And so I think it'll even out a little bit.
Thank you very much. Congratulations.
Thank you.
Okay, so Kelly, I'm convinced color is color. It's important. But if I said that complexity is more relevant than color, I am right or I am wrong from your point of view?
Wow, I don't want to pigeonhole myself. I think that when we are talking about large-scale projects and also the feasibility, I think complexity does play a greater role, mostly because some of the most important things when we're thinking about eco-engineering is providing more space and more space, just surface area to reduce competition, but also different sorts of areas for more ecological niches and functional roles. So I would say complexity is, but I would say that color also has an important effect, especially I think as we do more research, possibly for any species that are understudied or not always found in other areas or outcompeted, I think color might be able to help draw. We did do other sampling that hopefully will be analyzed later, maybe helping to support colors.
But maybe the color will change depending on the area, depending on the community, the habitat, etc. I was listening in the morning, I think, another presentation from this group. She said, the girls were saying that there are some experiments or something similar in UK, in some place in UK.
Yeah, that's part of the Living Seawalls project, and they're mostly looking at complexity and inserting different either panels or living boulders in different areas. So it's all, I think, towards the same goal of just improving our marine infrastructure, but depending on how we do that,
Are a company related with this group trying to produce this?
There's
trying to restore the boards, etc.
Yeah, I think right now it's more research institutions that are doing this sort of work in collaboration with private industry and corporations to build the concrete structures. So if there could be more of a tandem between that, then I think we could be doing a lot faster, get more done.
Okay. So, is there any experiment in that area related with the preferences of the invasive species or something like that? About complexity. What about that topic? Related with this.
Within my research, both that I did for my background and from our results, we haven't had many invasive species on our panels, and it's definitely not understudied. It definitely is understudied. If these are just creating more habitat for invasives, I would say yes, but I haven't seen any of the effects of any yet. So maybe we'll see with more time.
Some colleagues that are working in ports in Australia and New Zealand. In port, they have been doing a lot of things with the structure to see how that projected effect in ports help to the recruitment of invasive species and competition with native species. So I don't know if this topic is also related with this attempt to restore the habitats using eco-engineering.
It's more just trying to restore a part of the habitats that's lost, the color aspect.
You are in color.
Yeah. No, but it should definitely be studied from that perspective and point of view, because we don't want to help them. Thank you.
Thank you very much. It was really a very good presentation, very interesting. So it was the reason why I will ask you a very difficult question.
Okay. I'm ready.
And my question is that you state that there is an ecological value. I read what you write. An aquatic complexity into marine structure because it increases biodiversity. Okay?
Yes.
Do you really think it's always a good thing to increase biodiversity?
I do think it is important and good to increase biodiversity. If kind of going off that, if the biodiversity that's being increased is that of native species that might be being outcompeted in these flat, vertical, homogenized areas by creating more spaces for them.
I have an example which says exactly the opposite. What happened in Arctic currently?
In where?
In Arctic.
In the Arctic. With
biodiversity.
With biodiversity, well, I guess I'm going to assume it's decreasing. Because, no. Oh, it's increasing from a decrease of habitat. But by forcing...
Biodiversity in Arctic currently is increasing a lot.
Yeah.
And it's very, but the reason is that the polar bear is disappearing.
Yeah.
But in terms of biodiversity, we have much more species due to the warming. And new species are coming in Arctic.
Okay.
So the reason is that...
So changing basically of where habitats
it's the reason
have
of my question. Even, okay, it's the example of the Arctic, but sometimes you have natural environments.
Yes.
With low diversity.
Yeah.
And maybe it's not a good idea to increase the diversity because it will be another environment. It will be another ecosystem.
Yes.
I have many, do you have maybe another example where the biodiversity is very low, but the ecological value is very high? Do we have an idea in mind?
Yeah. I think I have ideas on the terrestrial. Like, this is something very interesting.
In terrestrial environment, but in marine
environments.
It's something similar to your situation, but no, you don't think about it, you have no idea?
Well, I know what you mean and what you're saying of how it's like shifting and everything, and why are we taking away a habitat that is going to lend itself to shifting biodiversity, whether it's in a positive or negative area, and it'll still support like a different amount, but I don't know of any examples necessarily off the top of my head.
An example is estuaries. In estuaries, the biodiversity is naturally low, very low. But the ecological value is very publication high. From Costanza, an economist, is very high. Because the productivity is very high. And so it's the reason, okay, maybe sometimes it's not always a good idea to increase the complexity of such devices, because maybe the natural diversity is low, and maybe it's
meant to be
it's a good use sometimes.
Yeah, I think that then could be something to further study, if this is like actually enhancing the ecosystem functioning as a whole and not just safeguarding species that had historically been there or not. I think that's mostly the goal right now. A lot of people still in denial of any large shifts to try to preserve what had been surviving and living in spaces. But I do understand that.
I said it is very.
I know, yeah. But it's making me think. I like it.
Thank you very much.
Thank you so much. Thank you.
So we still have I have one. I have some. So, have you control for surface? Because that's I don't know remember exactly the name, but the plane versus that one. I mean, the surface is.
The surface area is.
So, are you correct by that?
We are not correct for the difference in surface area. What we had just wanted was not to account for the surface area so much just as the area itself. So when we were doing the percentage cover, it was all across the same space of the panel because I just from, this wasn't something that I talked about necessarily with my supervisor, but it just to mimic the natural habitats, because when they would do censuses in normal rocky shores, they are just, once again, just putting the quadrant down and using that same space. So mimicking that sort of sampling and everything, it's just trying to keep it in an area regardless of the complexity of maps.
Okay.
Yeah.
So some question from the audience.
Yeah.
I have two quick questions. First, why is like the fourth panel like sideways? Is that like a factor, or is it just random how they were?
Whether some are vertical and some are horizontal. It was random. It was something that we noted down, but we haven't necessarily analyzed any large differences between the horizontal or vertically complex panels, but we did do it random so that we could possibly look at an effect. There's just a lot of factors at play, so for the purposes of this, we just focused on those. But it is going to be something that I look at, and from sampling, I could tell you that the horizontal ones definitely have. Yeah, they could just hold water at low tide more so. So there's more species usually in there.
And then are the color panels like much more expensive, or is it like the same for like if someone is going to build a whole wall of yellow?
Yeah.
Is it more expensive for them or not?
Moderately. So how they make it is once you have the oxides to create of the different colors, because they're in a ratio of the same four colors, just at different ratios. Once you have those oxides, it lasts a very long time, so you could build, ideally, a lot of different structures out of this. So there's not a large difference in cost between the colors and whatnot.
Just one last question.
Okay, I wanted to ask whether you've noticed like any kind of difference between like the organisms that were settling in the different color squares. In terms of, was there any kind of mimicking in terms of color? In terms of animals or algae, I don't know.
Yeah, we haven't seen any like consistent ideas of that, of any sort of mimicking from that. The biggest examples that I can think of is either our Hildebrandia watercipera that are red. We're not at higher abundances on our red panels, not yet. Same with our ova. The presence of that green ova australis was not dependent on the color so much, just as more so of how much light or shade those panels had to support it through the dry area. So not yet.
Okay, I think that's it. So thanks a lot. Thank you to everyone. So we have half an hour until the next session. We can Sorry for last year, and here appears 2024.
I never noticed 2024, and I saw the
I
dates.
think so. No?
Okay. It definitely happened to me once or twice because I
Yeah.
never realized it.
I mean,
But
I
now
think
it's
it's
just
still working.
For me, it doesn't matter.
Yeah, it's still working. I'm still getting the emails too.
Yeah.
I always wait for the email. Even they send the message, like, you should confirm.
Oh, thank you. Yeah, I like the things. I have
Okay, welcome everyone to this last session of today. I will be chairing this session. My name is Jose Robero from the University of Oviedo in Spain, and I will have a great company. Today, in the jury with Gonzalo Machado from the University of Oviedo, Spain, and Emilio Duvayo. I don't know if I pronounce well your from Garden, ITF, no? ITF, and Laura Mineta from the University of Oviedo. Okay, we will have 3 excellent presentations you will see. We will start with Catherine that will talk about tools. Okay, so it's your floor. Remember that you have 15 minutes. 1 minute before the end, I will stand up, so you saw that. Okay, enjoy.
Okay, hello, my name is Kate, and today I'll be presenting my research on the anthropogenic noise effects on cephalopod sensory systems and cognition. This project was supervised by Fabio Liotti and Katja Heugel, and was in collaboration with the biological station of Roscoff. Just a brief overview of what I'm going to be speaking about today. We'll discuss the rise of anthrophony, the importance of sound to marine organisms, and the interest of studying cephalopods and sound in particular. I'll discuss my project objectives, the methodology and hypotheses, as well as go through some preliminary and expected results. Since the Industrial Revolution, noise levels in the marine environment have been increasing. And we have been contributing to this through both intentional sound production, like sonar or air guns, and also through unintentional sound production, which are the byproducts of human activities like commercial fishing, or pile driving, or offshore wind farms. And unfortunately, I don't have a figure to show you or a map of how much sound we're putting into the ocean every day on a global scale. That's not being monitored at this time. But what I can tell you is that in the past 50 years, we've seen a 32-fold increase in sound levels along major shipping routes. And this increase in sound levels has major consequences for marine life. For animals that use acoustic cues to communicate, these are drowned out. And for other animals who rely on bioacoustic cues, like reef larvae, for example, to find proper settlement, they can no longer hear these cues. And in severe cases, sound can cause physical trauma and death in animals. But to better understand the complexities of sound, we need to understand how sound is different underwater. Sound is a mechanical wave that propagates through a medium by particle oscillation. And importantly, the speed at which sound travels is affected by the density of this medium, which is why, as you can see in water, it's so much faster than it is in air. When we compare this to both chemical cues, which are only relevant in high concentrations and at short distances in the marine environment, and light cues, whose propagation dissipates in the upper few meters of the ocean, it's easy to see why sound is the primary modality for so many animals, for communication, for environmental sensing, and for navigation. And the importance of sound is evidenced by this figure, which shows the vast number of animals who can hear underwater. But they don't all hear in the same range. And this has to do with how their hearing organs evolved. Some of their hearing organs evolved to hear particle motion, whereas others evolved more complex hearing organs, enabling them to detect the pressure wave aspect of sound or particle motion at the same time. And what's important about an animal's hearing range is what it tells us about its vulnerability to anthropogenic noise. I will be talking about, I'll be studying cephalopods, and the two species which are discussed in my research are Sepia officinalis and Octopus vulgaris. These two species of cephalopod are able to hear from 10 Hz to 1 kHz, so only low frequencies. And they're cephalopods, but they're also invertebrates. And in the marine, in the field of marine acoustics, invertebrates are severely understudied in comparison to fish and marine mammals. There's a huge gap in our understanding on how invertebrates can hear, if they can hear, and whether anthropogenic noise affects them. In fact, for Sepia officinalis and Octopus vulgaris, there are only 3 studies at all that look into the effects of anthropogenic noise on their physiology, and none that look into behavior. So why should we study cephalopods? Well, they are found everywhere, all over the world, at almost every depth, from hydrothermal vents to intertidal pools. And they're an important link in the food web, connecting small marine invertebrates and fishes to marine megafauna, to birds, and to Spanish. And what's really interesting about cephalopods is their cognition. They are incredibly intelligent. They're animals that evolved millions and millions of years ago from us. Our most common ancestor with cephalopods is a primitive flatworm. And yet they are able to solve complex problems. They've demonstrated both long and short-term memory. And so it's important to understand how sound might affect their behavior. They hear with 2 systems. The first are the epidermal hair cells. These have been compared to the fish lateral lines, and they're found on the arms and the funnel, as well as the mantle, and they enable cephalopods to detect local water movement. A little bit more sophisticated are the statocysts. These are paired organs. There's the macula statocyst statoconia, which enables cephalopods to detect both gravity and angular acceleration, and the cristocupula, which enables them to detect gravity and linear acceleration, and this is angular acceleration. Together, these paired organs create a really sensitive system in cephalopods for detecting particle motion and sound. And it's this sensitivity that makes them vulnerable to low-frequency noise. In a study done in 2011, Andre and colleagues subjected 4 species of cephalopod to really intense levels of low-frequency noise. And what they found is that the statocysts had severe damage. There was neuronal degeneration and swelling of afferent dendrites. And when these same levels of sound were applied to hatchlings of the same species, they also found lesions in the statocysts and damaged their epidermal hair cells. So we know that intense levels of chronic low-frequency noise can cause severe physical trauma to these animals. But what we don't understand is how lower intensity noise, chronic low-frequency noise, could affect their behavior and their development. And that's where our 2 studies come in. So we are looking into how ship noise playback can affect the problem-solving capacity of Octopus vulgaris. That's our first study. The second one is looking into how low-frequency sound can affect the development and the feeding behavior of Sepia officinalis. Now, both of these studies are still ongoing. We don't have all of the results yet. And because of limited time, I'm just going to give a brief overview of the first project dealing with Octopus vulgaris and trying to understand better how noise can affect their cognition. So we will include 18 wild octopus in our study, separating them into two groups. The control will be subjected to 110 decibels of ambient sound, and those in the noisy will be subjected to 135 decibels of boat noise playback. During these treatments, they will undergo a problem-solving opportunity, which is just a sealed jar with a plug with a crab inside that they have to extract. We're looking into how noise might affect their ability to solve these problems and to improve upon their times in solving these problems. Furthermore, we know that octopus have personalities, and we know that personality can affect their problem-solving performance. So we're including, among others, a neophobia test, in which we present the animal with a fake crab on a string and time its attack latency as well as approach, and compare that to its attack latency and approach to a fake crab on a string next to a novel object, which in this case was a Playmobil. With this information, we're trying to better understand how personality might affect their behavior under noisy conditions. With Sepia officinalis, we're doing something completely different. We're looking at the development and later the feeding behavior. First, we've taken 192 wild eggs, and we've separated them into two conditions. Again, noisy at 135 decibels and control at 110 decibels. The sound is supplied with these noise eggs that we made, which is basically just a small electromotor in a waterproof container that has a dominant frequency of around 175 hertz. They're incubated under these conditions, and we hypothesized, in the control we have the same noise egg, except instead of the electromotor producing the sound, we have a small resistor, so to enable us to control for the potential effects of the electromagnetic field. We hypothesized that eggs that are incubated under noisy conditions will hatch earlier than those in control, and that hatchlings incubated in noisy conditions will be smaller than those in control. Once they hatched, we moved each one into its own aquaria. We had 124 aquaria in total. And from the noisy, half of them went into control conditions, and the other half remained under noisy conditions. And control, the same. Half go into noisy conditions, and the other half remained in control conditions. Here, we're interested in understanding how noise might affect their ability to perceive and successfully attack prey. Each week, they will be presented with a different prey. The first, Paramesis, the second, Idotea, the third, Grangon, and the fourth, Quachius meus. And we hypothesize that the hatchlings and later on juveniles in noisy conditions will have longer attack latencies, and that they will also have lower feeding success than those in the control conditions. Well, I mentioned before, our experiments are still ongoing. We have concluded the incubation phase. And while we did not find that eggs in noisy conditions hatched earlier than those in the control conditions, our second hypothesis was supported. We found that those eggs, those cuttlefish eggs that were incubated in the noisy conditions, were in fact smaller, significantly so, than those incubated in control conditions. And this can be seen as a first alarm sign for the potential effects of noise on cephalopods. If it can affect their eggs, which are incredibly vulnerable, they can't move, they can't avoid the sound at all, creating stressful conditions resulting in smaller hatchlings, which may be more likely to be attacked by a predator, less likely to be successful at catching prey, and later in life, potentially mature later, have less reproductive success. This could have severe consequences on their biology. In conclusion, we know now from this study that chronic anthropogenic noise does affect the size of Sepia officinalis hatchlings. Hopefully, at the end of the study, we will know if anthropogenic noise can affect the feeding behavior of Sepia officinalis, and if it may affect the cognitive abilities of Octopus vulgaris, as well as if personality plays a role. But what we need for the future is a better understanding of how much noise we're putting into our marine environments, which starts with a better monitoring, or any monitoring, of sound pressure levels around the globe. In tandem with this, we need more invertebrate studies, because there's many things that we don't know, and if we don't know the effects of these sounds on the animals, we can't anticipate the effects that the growing number of our global fleet might have. Furthermore, I suggest that once we better understand these things, we can push for policy intervention, starting with the requirement of quieter motors for commercial vessels. Thank you.
Thank you so much, Katarina. So we have some time for questions. So Laura, can you start?
Okay, yes, for sure. Thank you very much for the presentation. I was quite surprised about how octopus and Serbia here.
Yeah.
I don't know that. So, but I have some technical question, like for the design of the experiment.
Yeah.
You say that for octopus it was the vessel noise.
Yes.
And the other one is low frequency.
Mm-hmm.
Why these two different types of noise and not the same for both of them?
Yeah, they're actually both low frequency noise, so vessel noise is low frequency noise. But for the octopus experiments, we're using an underwater speaker because the tanks are much larger. Whereas for the sepia experiments, the tanks are about this big. Mm-hmm. So we had to make these mini these mini noise eggs in order to have an underwater speaker of that size.
Mm-hmm. Okay, and I see that one of your recommendations for future is to have engines with less noise. Mm-hmm. Do you think that this is feasible or is possible? Because at the end in land surface we have how do you say, electronic engines. Mm-hmm. When you have a car with electricity, you don't hear it.
Yeah.
Yeah. So maybe if this could be a solution.
It's possible. I mean, for a commercial shipping vessel, I'm not sure if fully electric would be possible at such a large scale, but I know that part of the reason why these vessels make so much noise is because the motors are inefficient. So I think it is feasible because there would be even commercial value in making better, more efficient motors.
Mm-hmm. And yeah, I think that's it. Thank you.
Thank you.
Thank you, Laura.
Yeah, thanks very much. Nice presentation, well done. I have a couple of questions, but one was, how did you come up with actually there's data right there, so it's easy. What did you base these hypotheses on, that the eggs would hatch earlier and that the hatchlings would be smaller? How did you come up with these?
Well, we know that stress for sepia, like heat stress, for example, will decrease the time it takes for them to hatch, and sometimes decrease the size. So I guess I was thinking of noise as a similar kind of environmental stressor as temperature.
Do you think noise and temperature would act the same on eggs? Like the kind of physiological, like what is it that's affecting the eggs?
Yeah, I think because it's like it is a physical movement of particle motion. I think it's kind of similar to temperature.
Yeah. Okay, thank you. And then the other one was, you mentioned about the personality and adding the novel object in the tank. Can you expand a bit on this again, just on what you based it on? Was it prior research?
Yeah.
Has there been other studies showing
the
effect of the novel object?
Yeah, so neophobia tests have been used for lots of different animals, but they've been particularly used for octopus in assessing their personality. And by understanding, like, this is a pretty textbook timid animal. You can't see the speed at which it's attacking the fake crab, but you can see that it's reaching out with one arm, like it's shy. And these are reflected in their attack latencies. But this is just one part of our understanding of their personality. We're also have an ethogram in which we track their behavior in multiple different settings and different contexts, because that's what personality is. It's the same behavior over multiple contexts across time scale. So we take all of that into consideration.
Okay. Yeah, I think the only other clarification would be going back to the ship noise.
Yeah.
So you call it ship noise, but I'm wondering what is it really and how is it relevant to the octopus?
Yeah, it is actually, it's generated ground noise, which is similar to ship noise. And it's relevant because these animals, the area from which we took them was in the harbor where the ferries pass in Roscoff to go to Ireland, like big boats. And we got these levels by deploying a hydrophone for, I think it was 10 days, and the ambient level was 110, and when we had boats passing, the noise levels went up to 140 decibels, and this was at 25 meters of depth. So we chose these levels to reflect those kinds of, the experience that they normally have.
Great. Thanks very much.
Thank you.
Thank you, Federico. So thank you, Catherine, for such a nice work. Oh, it's preliminary work, but still it's a work in progress. So well done, and those manipulative experiments are not easy. There's always some things to adjust, so it's complicated. So one thing is, just remind me about octopus biology. So you test eggs, separate eggs, not aggregate eggs.
Separate eggs.
So separate.
Yes, but those were for cuttlefish.
Sorry?
Those were for cuttlefish with sepia.
Okay, with sepia. Okay, with sepia. So, but the thing is that you test separate. But there's in nature, I mean, normal reproduction biology, so the aggregation of those.
Yes.
So you expect that might affect noise might affect differentially depending on how they are aggregated. Have you planned to test that somehow?
We do have them, they're separated individually, so they're not stuck to one another like they are in the wild.
Yeah.
But we do have them in baskets. So for each condition, we had 3 replicates, and in each of these tanks, we had a small basket in which the eggs were actually aggregated together.
Okay.
Yeah.
Okay, to test that difference, that could be also interesting.
Yes.
First, if you find separate, but then see if depending on which position you are, then when they are developing, if the performance is different. So, and also in terms of this is with octopus, in terms of there's some sort of standardized test for boldness. Because for fish, for example, clearly you have these circles and you count the amount of time that they go from, from dark circle to the white area, and there's some standardized. Is something similar for octopus?
There has been a study done by Matza in 2022, I think, in which she made a standard battery of tests to determine the personality. So that's actually where we took the neophobia tests, and why we're recording their different behaviors across different contexts. So we record their behaviors twice a day during welfare checks in the morning and in the evening, also during the neophobia tests, during sound trials, during the control trials.
So completely random, I guess, and also the order, you switch the orders.
Yes, of this we switch the orders. We have 3 trials in the morning, we have 2 trials in the morning, 2 trials in the mid-morning, 2 trials in the evening, in which we switch the order of presentation of the crowd.
So there's some sort of learning.
Exactly.
And this kind of
Exactly. Yes.
So, I think I don't have many more questions. So thank you very much.
Yeah. Thank you. Thank you.
Thank you, Catalina. We have some time for more questions. I just want to ask: are there octopus in Roscoff?
Yes, there are a lot of octopus in Roscoff. Oh yeah. Yes. Right now, yes.
Because we were working on that with some people, and they say that before, a few decades ago, the population was huge. But now, or 4 years ago, it was scarce.
Yeah, there was a big population boom in Roscoff, and then they started to eat all the coquilles Saint-Jacques, and so the fishermen were upset. The fishermen started to fish all the octopus. They disappeared, and now they're back. People think it's because of the warmer winter, maybe. but
Interesting.
yeah.
Yeah, because we were working with some people there in the school, at the same time we were working in barnacles and in octopus, and they said that at that time that they were not octopus. I mean, it was a few years ago.
Yeah.
Another thing, are there any evidences, are there any evidences that could relate the decline stocks in some fisheries or whatever due to the increase of sound pollution in other species?
For fish specifically?
Yeah, for example. You said that in vertebrates there are almost nothing. It's almost nothing.
Yeah.
But in other species?
I don't have a lot of knowledge on how it affects fish. I didn't really study much of that for my review. Sorry. But they do have otoliths, so they're able to perceive particle motion, and many of them can hear actually pressure wave of sound. So I think that sound would definitely affect them in some way.
Okay, we have some time for questions.
Yeah, I was thinking that since you have this slide here, if you take into account females and males.
We don't, because in the past that hasn't made a difference in personality, female and male. We do record if they're female or male, but in the personality analysis, we haven't been taking that into consideration.
Yeah, but maybe for noise audiences.
Yeah, it's possible. Who knows? We'll see.
Okay.
Yeah.
We have a question there.
Okay.
From the audience.
It might be a very silly and non-important question, but I would like to ask why did you choose to create like different studies depending on the species? Does it have to do with the curiosity about a specific species, or is there any other reason?
There are multiple reasons. One was just logistics. Getting the permits, the ethical permits for these things takes a really long time. That's one of the reasons why we're so behind in the experiment. So we wanted to have two choices just in case. But also these kinds of problem solving, at least with jar tasks, are much easier with octopus, whereas with cuttlefish, they're less dexterous. They have been able to just solve some problems, like mazes, but yeah.
Thank you.
Yeah.
Yes.
I'm interested also in the neophobia test, because you mentioned that you did it with a fake trap, right?
Yes.
And octopus, right, they have quite good eyesight, but they also feel receptive. Do you think that maybe the fact that it's a fake trap, it doesn't have the same chemical signals as a real trap, do you think that might affect the behavior of the octopus, but maybe it doesn't recognize it as a prey, but as maybe a toy?
Yeah. No, that's a good question. There's actually been a whole study because people are worried about the ethics of using live crabs on string and exposing them to in front of a predator, multiple times. It's really stressful for the animal. So there's already been a whole study examining whether false crab on a string is an appropriate way to determine the attack latency of the octopus. And they found that yes, and we're finding the same thing, that as long as it's moving, they're interested.
It's moving as a shape like a crab.
Yeah, pretty much.
Interesting, thank you.
Yeah.
Another question? Yes.
So, like, you have the control and then you have the other ones under noise conditions. So just to know, or is it like constant noise at the same frequency, or would it be like change frequency and all of this?
Yeah, actually, the sound is only being played during the problem-solving trials. They have 30 minutes to sort of not fully habituate to the sound, but so that it doesn't startle them and change their behavior. But apart from that, they're in this 110 ambient level of noise.
In this kind of tests, is there something correct about social relationships? I mean, it's only that kind of test of personality or problem-solving experiments are with only one octopus in one time, or there's something about social relationships?
Well, octopus are naturally solitary, but they can learn from their conspecifics, which is why we positioned all of our tanks so that they can't see each other, because they can learn really easily from one another. So Yeah, it would be interesting, but they don't really like to be with each other.
We have one time we found a file of topos and it was crazy.
Ah, really?
Yeah, because we were doing some environmental DNA analysis and we wanted to correlate the amount, the total weight of topos, so we have several times with different quantities of the topos to quantify.
Oh.
It's crazy.
Yeah.
Okay, thank you so much.
Yeah.
Okay, so now we have Ariana. Are you ready?
Yeah.
Go ahead.
So, welcome everyone. I'm Ariana Bett, and this is my thesis project, optimizing bottlenose dolphins research, performance comparison of 5 passive acoustic monitoring devices, and data processing recommendations. So, first of all, what is passive acoustic monitoring? It's a non-invasive technique that uses underwater devices to detect and analyze sounds in the marine environment, including vocalization from marine mammals. Marine mammals use sound to navigate, to communicate, to locate prey, but they're not, of course, the only source of sound present in the ocean. The ocean soundscape is characterized by 3 components that interact with each other. The first one is biophony, and it includes all the sounds from marine pets, so for example, mammals, fishes, and invertebrates. Then we have the geophony, that includes all the sounds from geological sources, such as waves, rain, and volcanic and seismic activity. And then we have the third component that includes all the sounds from human sources, such as shipping, drilling, military activities, fishing, and it's the one of increasing concern right now. So, to study this complex soundscape, a lot of pan technologies have been developed, and between them we have the autonomous acoustic recorders that are especially useful because once deployed at a specific depth, they can independently record for long periods in remote areas. They're ideal for remote areas and critical species, and they store data internally until recovery. So, in the market right now, there are over 40 autonomous passive acoustic monitoring devices, and so it's important to say that they differ in type and technology used. So, to avoid this data misinterpretation and allow to compare their outputs, it's important to compare them. So, in this study, we decided to compare 5 autonomous passive acoustic monitoring devices, and they were tested especially for the ability to detect bottlenose dolphins vocalizations in the context of occurrence studies. So, these devices belong to the 2 main categories of passive acoustic monitoring devices: sound recorders and click loggers. Sound recorders capture a wide range of sounds, biotic and abiotic, from biotic and abiotic sources. They store raw data and require post-processing with software like Raven or Panda, and they're very suitable for data analysis because you have a complete view of all the sounds present in the ocean at that moment. But this means that it requires a lot of battery capacity, and also large data storage. On the other side, click loggers are specifically suited to detect echolocation clicks from odontocetes. So they do not store raw audio data, but they just collect information related to the clicks. So, for example, the click timing and peak frequency, and then these clicks are grouped into click trains through a software that is proprietary of the manufacturers of the click loggers, and is FODEX with the kernel detector. They have, in this case, lower power consumption, and they require smaller data storage. So these are the devices that are used for the price in sample rate and in the cycle, and in type as present. So the ultra mode is the only one that is
the low-cost device, and low-cost devices have been developed right now because usually passive acoustic monitoring is very limited by cost. So this is one of the attempts to develop a low-cost device. And C-pod and F-pod are the flip-floppers, and F-pod is the new version of the C-pod. So the study area was the Shannon Estuary, is a special area of conservation since 2000, is in West Island, and the devices were co-deployed for 2 months at 12 meters depth. And this estuary hosts long-term bottlenose dolphin population with high site fidelity and year-round occurrence. And this area was a good setting for this type of comparison because it hosts only one delphinid species and they're always present, so we could have a controlled comparison. The analysis was divided between whistle detection and clip rate detection because of the type of devices used and their sample rate, and because, of course, of the species considered. So for the whistle detection, these are the 3 types of devices considered, and we use Panguard as a software. It's an open source platform, and specifically we used a tutorial that was on the website to make the analysis replicable by other users. So we followed this tutorial following all is setting only for the soundtrack because it was only the one that closely matched their sample rate, the one described by them. For the other 2, we had to adjust the spectrogram, changing the parameters of FFT length and hop size, because the whistle-on-one detector works as extracting figures from the spectrogram that is used as an image. So we had to have the same image with the same frequency and time resolution for all of the devices. Then we processed the data using Panguard, and the output was converted into detection positive minutes per hour. This basically means that for every hour we have the number of minutes that have at least one detection. So these are the results. As we can see, even if they were co-deployed, they show very great differences. It's a 5 days time series map. 5 days was limited, the number of days was limited by the battery capacity of the other mod, the low-cost device. And so in this graph you can see that none of the devices recorded 0 detection. So for every hour we had detections. So this is just a very unrealistic situation where we have dolphins always vocalizing and always present. And also if we see the percentages, very high percentages of detection positive minutes and very low or no correlation between the devices, even if they were co-deployed. So looking at the spectrograms, we could see that the whistle-on-one detector was able to detect the whistles, but it was also catching a lot of noise at low frequencies. And this could be due to the fact that we are in an area with very high anthropogenic noise and ambient noise, because it's one of the main deep water ports in Europe. So we had a lot of false positives. So we decided to try to improve the accuracy of this detector using a high levels of anthropogenic noise, and we tried to improve the accuracy of the detector using a context-dependent parameter. So we annotated in the recordings a subset of whistles. We collected the duration, and we took the 10th percentile of the duration, and this value was used as the new minimum duration for the detector in order to try to exclude all the noise. So these are the new results. All the devices decreased in their detection positive minutes, and also the premium increase between devices, especially between the soundtrack and the RTZs that are the ones that were recorded continuously and are the ones that are not low cost, so they're supposed to be better at recording. And if we look at the spectrogram, we can see that we still have the whistles, but we got rid of all the false positive, all the noise at very low frequency. And so this improved accuracy, of course, comes with a trade-off, because
in this case we are selecting, we are putting a filter on the duration of the whistles, and so we have more accuracy, but maybe real whistles that are very brief could not be detected. This was to show that it's important to tailor the settings of the detector to improve the device's reliability. We also did a manual validation of the two settings, so we took 3 days and we annotated all the whistles for the 3 days. We can see that for all the devices, the accuracy improved with the new setting. The new setting is in red, the blue are the manually annotated whistles. And as remote, despite its low cost, was performed reasonably well. And regarding click detection, we had a completely different method because in this case we were using devices that were both click loggers and both sound recorders. So for the F-pod and C-pod data, the devices were, the clicks were grouped into click trains directly in the software, as you can see here, and the software has grouped them and assigned them to a source category like narrowband high frequency localization, all station, toner, and unclassified, and a quality class, high, moderate, low, which reflects the likelihood that that click train actually reflects a true click train. For this comparison, we only consider the other station category because it's the one to which bottlenose dolphins belong to. And for the quality trains, only all the less restrictive grouping, so high, moderate, low trains. And the data were directly exported as detection positive units from the software. For the soundtrack, we had to a different approach because we are dealing with a sound recorder. In this case, we used Vanguard again, and in this case, we use the soundtrack click detector model, but as we can see, we have as an output clicks, but are not distinguished between sources, not biotic or abiotic. We don't know the source of them. So since we are in a high anthropogenic noise area, we had to filter them, and based on the literature, so we select which ones were the clicks from the bottlenose dolphins. We retained only those, and we grouped them manually into click trains, considering a train as made of 5 clicks minimum, with an interval of maximum 300 milliseconds. The output was also in this case detection positive minutes per hour. And in this case, we have to say that this was a trial because the algorithm that's used in the software of the click logger is proprietary, so we don't know exactly how they extract the click trains from the devices. So we had to try to compare the outputs of the 2 devices, of the 3 devices, but of the 2 categories with trials, basically. So these are the results for 5 days. In the blue is the soundtrack and green is the F-pod, and we can see that it showed good agreement. And in this case, we only are representing the click trains with like the F-pod, the click logger setting with the setting high, moderate, low click trains. So with all the trains with all the qualities. So we can see that the soundtrack was agreeing with the F-pod when the least strict filter was applied, and if we also see the correlation, no correlation was found between the C-pod and the F-pod, or between the C-pod and the other the soundtrack, and this is very important because if we consider that the F-pod is the new version of the C-pod, if we go to the field and we replace the C-pod with the F-pod with not careful evaluation, we could have differences in detection rates, but they could not actually necessarily reflect differences in animal activity, but maybe just a difference in the device used. So we have to account for that. So just to conclude, this study provides a preliminary comparison of 5 passive acoustic monitoring devices for monitoring bottlenose dolphins occurrence, and customizing detector settings is important. we customize them based on the area in order to have reliable data. Soundtrack was the device that offered the richer data because there was a combination between sound recorder technology and click detection. iDreamOt showed to be a promising device, even if it's a low-cost device, especially for understudied regions or remote areas. Click loggers are useful, but we need to take into account a possible presence of bias due to device replacement or due to the algorithm they're using. And for future studies, it would be useful to compare devices with the same sample rate and duty cycles, and also we should consider the effect of noise on the detection rates of the detector. And this was just finished. I used to call detectors just not a very sensitive detector, but a detector where there is a suitable trade-off between misses and false reliers. Thank you.
Thank you, Anna, for your presentation. Okay, so we have time for some questions. Don, do you want to start now?
Yeah, so congrats. Thanks for such a nice presentation. So, I mean, this is far beyond my expertise, but in any case, I think it's super interesting. And this kind of calibration or inter-device difference assessment, I think it's quite relevant, since what you were pointing out at the end of your presentation is that if for long-term studies, if you suddenly change without proper understanding what you might be missing, so I think those are really important. So just to highlight even more the relevance of your study, that I think this kind of study are necessary. Were some studies similar to this done before in other areas with other, allow me to say, with other anthrophony or geophony in different parts of same region or even different region?
Yes, especially there are a lot of studies that compare the two categories. I'm just gonna put the slide again, so in case. Sorry. These two, the sound recorders and click loggers, so F-pod, C-pod against soundtrack, for example, because since they work so differently, of course we're trying to compare them, and also between C-pod and F-pod since they're of the same manufacturer. And regarding C-pod and F-pod comparison, there was not a study that actually found agreement between them, but in Ireland, but in that case they were focusing on purposes, and in that case it's another category in the software. It's a narrowband high frequency category, so it's probably the algorithm works differently for purposes detection and dolphin detection. So that's what just a parenthesis. And regarding noise, I know that there's they did a study in Denmark, for example, especially on the effect of noise on their detection rates, and they saw that the devices are affected by noise. So some of them, maybe if there is a high level of noise, they're gonna detect more, and others not really. So we have to account also.
Because I would think if you will find something different because you were like reading the page,
Yeah.
if you move a little, so if that might change your result or not, depending where you are located and the geography and how that has an effect in noise dispersion and something like that. So the other thing is, so the way that, correct me if I'm wrong, I'm really naive about this, so I'm not an expert on that. But the way that you are extracting is automatic, so you get the record and it's automatic. And you are somehow, you are filtering, you apply many different filters to the text. Is there any way, because I mean, related with the way that from a previous thesis, that they were doing identification of sharks based on deep learning. Can you apply deep learning, or this I was just looking at it, is there any deep learning to, because you can train with a mix of different noises, even from the same anthrophony or geophony, with a mix of noises to see how good they are picking up your particular species?
I think it would be really possible. I think this software in particular, Vanguard, is implementing now a new model, trying to, of course, adopt deep learning because it would be much easier. For now, it's just automatic based on the contours that is used on the spectrogram. So yeah.
Because even if you could have some of this intercalibration depending on the general conditions, so you, if you apply one device to the other, can make some sort of corrections to get still detecting the particular species that you're focusing on. So I think your work is really relevant for this matter. So
thank you.
Excellent. Well done.
Thank you, Rob.
Thanks very much for a nice presentation. I was wondering actually why you have the slide open. You mentioned that with these clicks that you have the background noise and so you might be missing some sharp clicks, but it looks like the frequencies are quite different. So why would you be worried about missing?
No, in this case, it was about the whistles. And so since I'm considering, so now we have all this was the default setting, so not changed. So we are, I don't know if you can see, it's taking a lot of noise here in blue, and it's considering it as whistles, but they're not. So we decided, based on the settings you can change on the software, to try to put filter on the duration of the signal. And since they're very brief, we thought that would work. And so if we put filter on the duration, of course, if a whistle is very small, it's not going to get.
Yeah, but you see, I'm looking more at the y-axis than the x-axis. On the y-axis, the frequency is very different.
Sorry.
So why would you just not cut the point on the y-axis?
Because, I also, at the beginning, I also wanted to do that. It's the more reasonable way to act. The problem is that on the software, for this type of model, the whistle moment detector, you cannot select before, like we did it, we process the data, we don't select them after. So you cannot put anything related to the frequency in the...
Okay, you can't change the range.
I can show you the... I don't know if you can not select this one.
I know what you mean.
So, I don't know, here, I don't know why it's not, but there is like, these are the parameters that is possible to adjust. The minimum length, the minimum total size in pixel, and there was like the crossing, maximum crossing or something like this that is related to the spectrogram feature. But
because that seems like an easy way to just take all this immediately. And then kind of related to this, and also this is definitely not my area, so this might be a very quick answer, but you talked about the biophony and anthropophony and geophony, but what are the frequencies of these? How do they overlap? How are they different? What's the signature of the anthropophony, for example?
For example, here, like the anthropophony is very variable as frequencies. For example, here we can see that there's like a line that probably is a boat that is far crossing. Or, for example, these clicks, this line could be, for example, sediment that is going against the hydrophone, that is like hitting the hydrophone. And for when we have shapes, the frequency, like this is the frequency label, axis on it. For the ship, we have a high variety of frequency ranges.
So we can't really look at something like this and separate geophony, biophony, and...
not that I know of. More based on the features, so how they look like rather than...
And then you have to go back and figure out which is which. Okay. And then the last question is kind of still a little bit on this. So you said that you sharpened this to remove the ambient noise. And then you mentioned at the end that it would be interesting to look at the performance under different noise conditions. But do you have that data? Can you use your ambient noise?
I can. Like in the future, that was probably something that I could do, but I didn't have time to do that.
So you can look at your detection rates with your ambient noise. Okay.
Yes.
And you expect to see?
Yes, to see differences based on the territory. Yes, because there were some, since we are in a deep water port, there were also moments in which the spectrum was completely like black because of very noisy ships that were passing near the hydrophones that are all co-deployed. So, so it means that we have a very huge variability in the noise in that area, even if we have one station. So we could we could study the difference in detection rates. But it would be, I think, also useful to have more deployment stations to compare. Also more devices. Yes. Thank you.
Yeah, thank you.
Okay, thank you so much. Laura?
Okay, congratulations, it's done. And I enjoy a lot the presentation. And I agree with Gonzalo that these kind of studies are very needed because when you are planning to buy one of those devices, you don't know that there are so many differences between one to each other. And particularly in these studies, just focus in these species of dolphin. But when you are looking for any, this is slightly different, and particularly if they are rare species, is the difference between detected or not detected. So I think that it's really very useful. And my question is very related with that because the studies focusing with the dolphin, why only that species and not any that is around?
Because in the Shannon estuary, we only have bottlenose dolphins. We have occasional presence of porpoises, but the thing that the devices that we were using had sample rates that were not able to detect the vocalization of the porpoises because porpoises have very high frequency vocalization, so we need very high sample rates. And in this case, the majority of the devices could not detect it. And also because the click loggers only detect odontocytes, so we could just use that species.
And do you have, during the study, other ways to detect if the dolphins are present or not, like visual or?
Yeah, like this data were collected in 2024, so I didn't go in the field, but yeah, we could actually do that. They did it in previous studies, both visual sampling combined with acoustic monitoring.
Yeah, particularly to see if they are correlating. The dolphins are there, we detected or not.
Yeah, a study was done where they were measuring the detection range of the hydrophones, and in those cases, it's important to see like how far is the animal to see what's the detection range, because that is something that could affect the differences in devices.
And you already go to my next question, and it's about the detection rate.
Yeah. Like
how far you are able to detect it, and if they are different between devices, because maybe that's the difference that you can see.
Yeah, very recent studies, a study by Todd and colleagues, showed that, for example, the difference in between sound trap and F-pod, sound trap, F-pod and sound trap related to the C-pod could be the detection range. So it could be that this new sound trap, the sound recorder, and F-pod, that is the new version of the C-pod, could have a bigger detection range than the others. In general, the sound recorders don't, like the manufacturers, for example, don't specify the detection rates, but because it's very variable and based on the study area. And for the C-pod F-pod, they I don't remember it exactly. I had it, but yeah, they specified that it's around 300 meters for arbor purposes and more than 1 kilometer for dolphins. Yeah.
And maybe a future study is to how far.
Yeah.
Okay, thank you very much.
I was reading in the past that are you able to detect how many individuals or specifically detect an individual with this kind of devices or not? I mean, is there a correlation or this can even go to an individual identification because of the sounds?
In this case, we didn't focus on that. It was more like presence presence of the dolphins, not on the number of individuals, but we could certainly do that. I don't know if this method could work for that because we are excluding some brief whistles that could be whistles. So in that case, we should develop another way to exclude all the noise. But yeah, it would be possible to.
How long was to modify the default parameters? That was right for how.
It was very long, first of all, to figure out how this software works and to make it work because it's tricky and not very intuitive. But then, yeah, and then it gets a lot of time to process the data. They're not long. It was just 1 month more or less, and the other one just 5 days. But it takes a lot of time to process them, and yeah, I think I figured out like the customized setting after months and a half, probably. Yeah. Very long.
Okay, so we have time for one question from the audience. Anyone? No. It's done.
Thank you.
Last but not least, we have Mario. That will talk about weight. Are you ready? Are you okay? Yes. Okay.
That 15 minutes.
Okay. Go ahead. The floor is yours.
Hi everyone. I will present you a deep dive in the male sperm whale ecology in the North Atlantic. I did a literature review with an acoustic case study in the Norwegian Sea. Despite that this species is well known, most of the studies are focusing on tropical and subtropical females, and much is still need to be understood on male sperm whales. I conducted a non-systematic literature review on several databases of peer-reviewed studies like Web of Science, Google Scholar, and Semantic Scholar, and using as well Grey Literature. And after a screening process, I reviewed 297 papers on male sperm whale ecology in the North Atlantic, focusing on the distribution, habitat preferences, abundance, seasonal migration, their feeding ecology, their population and social structure, and mostly the anthropogenic impact on this species. I conducted a case study doing a visual and acoustic survey within the Norwegian Sea, in a potential deep sea mining area designated by the Norwegian government. So the aim was to identify the critical knowledge gap and to contribute to the knowledge within the potential deep sea mining area for better conservation mitigation. So, sperm whales is a cosmopolitan species. They are sexually segregated. They live from equator to pole. The males are distributed up to 89 degrees north, and no female can be found, or at least haven't been found yet, up to 50 degrees north. And they live in every ice-free water. They're mostly associated with eddies and upwelling system, as well as with temperature gradients, and they favor complex geological topography like ridges, canyons, slope, trench, etc. in waters deeper than 1,000 meters and going up to 4,000 meters. Until recently, not much was known about male migration in the North Atlantic. With some harpoons evidence and photo identification, we had some information on connectivity between the Azores in Spain and the West North Atlantic Ocean, like Gulf of Mexico and Bahamas. But recently, a paper just came out this year with 29 tagged male in the Norwegian Sea. 12 of them migrated south. It gave us a lot of really important knowledge on this species. We know now that they don't really have a fixed breeding season because they displayed asynchronous departure from January till October. And they spread it in this very large area close to the equator in the North Atlantic of over 10 million square kilometers below 45 degrees from their feeding ground in the Norwegian Sea till their breeding ground close to the equator. So male sperm whales are feeding mainly on cephalopods, but they are generalistic predators, and their diets differ from within the region, within the North Atlantic. They can dive between 45 minutes and up to an hour in depth between 400 meters and 2,100 meters. They're using echolocation clicks to orientate themselves and forage and shift to buzzes or tweaks that are like very high frequency clicks to give them high resolution at the end of the prey capture moment. We know now they switch between different foraging modes to select foraging strategy based on energy optimization. So for shallow mid-depth where preys are more mobile, they have like a way more active click echolocation vocalization, while for deep dives they save their energy and have and use less click patterns for less mobile preys. Sorry. So sperm whales have been hunted for centuries, and the population declined pretty much for whaling due to whaling between the 18th and the 20th century, and didn't really recover from that, and they are now facing a lot of current threats like pollution, both microplastic and noise pollution. Microplastic can injure the gastrointestinal tract that can result into death sometime, and noise pollution can be an important threat to sperm whale and alter their feeding behavior and their orientation. This species is heavily impacted by vessel strike. However, males are underreported because living in more deeper and offshore waters, and most of the reports are on males and females. They suffer as well from an intense fishery pressures and the interaction that can result into entanglement, bycatch, and also depredation behavior that will also alter the feeding behavior and prey capture attempt. Climate change can also have impacts on sperm whales. It is not clear yet if it will actually benefit the species, have no consequences, or alter their prey and their prey distribution, and then their food availability. Finally, an emerging threat coming, deep sea mining, that is expected to have operation 24 hours a day, resulting in noise pollution up to 120 decibels for several kilometers around, and up to 90 decibels for hundreds of kilometers radius. To implement a good conservation measure
some knowledge gap needs to be fulfilled. About male migration, there's still a lot to know, and we still need to identify male migration corridors to protect the critical habitats implement conservation measures in those critical habitats. About male social population structure, not much is understood in the opposition, contrary to female social structure that are well studied. We know now that there's gathering happenings in the Norwegian Sea, up to 12 individuals during feeding events, and there's probably a seasonality with that, but it's not completely understood yet. We also know that they, as female, produce social vocalization like codas or slow clicks. However, they still need a lot of investigation on that. There is a lot of unknown. With the intensification of anthropogenic threats now, we still need to know what will be the consequences on male population. Fishery pressure is intensifying now, and depredation can have an impact on male behaviors, and probably also an impact on the migrations. However, there's still a lot to study about that. You can see that over the years, vessel strike events on sperm whales increased pretty intensely, but as I said, they are mostly unreported for males. Climate change will probably also have an impact. We know that it also already have an impact on the distribution, but however, we don't know about the food availability, and there's still a lot to know. Anthropogenic noise, we know that in some circumstances it is actually having an impact on them, but it's depending on the source, the sound duration, the frequency range, the sound level, and the distance from the source. And some experiment didn't show any response from sperm whales, whether some others showed pretty intense avoidance behavior, stop information activities, etc. But because human is not learning so much from a mistake, we're adding more stress to already stressed environments. As I said, sperm whales' sensitivity to sound is still unclear, and we will need to investigate more the behavioral impact on feeding and navigation abilities on male sperm whale before conducting any industrial operation, and to assess their sensitivity based on sound, energy level, frequency, and distance. Sorry. It is actually a huge knowledge gap on the impact of possible large-scale commercial operation of deep sea mining that are starting to be planned now. And considering the already stresses in the environment, it will be necessary to evaluate what we will add in those environments before adding any other extra noise pollution, and to develop complete understanding of the consequences of this industry. This is why we conducted a case study in the Norwegian Sea. So the Norwegian government dedicated 280,000 square kilometers to potential deep sea mining in the Norwegian Sea. This is where we're conducting our visual acoustic survey from 30th of July to the 10th of August 2024. We used a tone hydrophone array to conduct a passive acoustic monitoring survey, and all the sperm whales encountered were manually marked on barcodes.
So out of 211 hours recorded over more than 2,700 kilometers, we encountered 22 sperm whales, 18 were within the deep sea mining area, and 9 of the encounters were groups up to 3 individuals. Most of our encounters were during feeding of these animals, so we can assess that this is a really critical feeding habitat for male sperm whales. So deep sea mining, we know now that it can alter the biochemical environment, the food web, and add a lot of noise pollution to the environment. And we know as well that we know as well that in the Norwegian Sea, this is a really important feeding habitat. As we saw on the migration map, the Norwegian male sperm whale population is migrating to a very large area in the south and is probably to breed and is probably supporting genetically a lot of the North Atlantic sperm whale population, and an extra noise pollution in this area will have a population level effect and conservation implication. So now, looking at all of that and what we know and all those knowledge gaps, we definitely need to investigate more on male sperm whale migration patterns. There's a lack of systematic data sharing system for photo identification, which is a shame because we know that there's a lot of photo identification data that are not analyzed. So using shared repertories like Happy Whales would help scientists to have join their efforts and have way more information on connectivity within the ocean. We need as well to investigate seasonal pattern in migration and also social gathering in males in high latitude that is completely unknown now. We definitely need to have a better understanding of our human impact on sperm whales before adding any other extra threat to them, and to assess the potential of this emerging new industry. So to apply the precautionary approach to marine marine development to avoid any extra threat without knowing the impact, and we need to advance scientific understanding of this population before any industrialization of our ocean use, and establish safeguards before expanding the industry. To conclude, I was in the Arctic 4 days ago. We keep having collecting data in this region, so I'll be very happy to answer your question about that. We have a paper, team has a paper that came out yesterday on the threatened cetaceans in general in the CCZ zone in the Pacific, which is a critical zone for deep sea mining right now. If you watch the news, it's pretty bad. So if you want to have a look at that, I think it's pretty relevant. Thank you for your attention.
Thank you, Mario.
Yeah, no problem. Thanks a lot, Marianne. Really nice presentation. Your case study was interesting and well outlined. I was curious what you got from the review. So you said you did an unsystematic review with 297 papers. Did you have some main conclusions from this, or what was the main?
The main conclusion is that there's like a lot of knowledge on female social structure and behavior, and it's like well studied and it's really attracting new studies and everything, and there's like basically nothing on the male, and also on the genetic connectivity is missing a lot, and migration patterns is also missing.
Yeah.
And was that for this area in particular or for global?
For the North Atlantic,
Okay.
mostly, I mean, a bit more is known on the Pacific. Studies have been made more on the Pacific. However, for males, it's still really a lot of unresolved questions. Yeah.
Because I've heard about allegations of non-breeding teenager males, like for example in Kaikoura in New Zealand. Do you think there is something like this, that this male population is a population like that, or?
Sorry, what do you mean by that?
In Kaikoura in New Zealand,
yeah.
They're thought to be non-breeding teenager males that hang out together.
Yeah.
Is that something that's happening here in this
Yeah, so in the North Atlantic there's also this thing about bachelor groups where they start going in higher latitudes before being sexually mature, and once sexually mature they just have this solitary lifestyle. Yeah, which is similar from what you told me.
Is that what's happening in this group?
Yeah.
Okay.
Yeah.
And then you outlined a range of different issues or effects on these, and focused of course on deep sea mining, but I wonder, is renewable energy also an issue for sperm whales?
Sorry, I didn't hear.
Is development of renewable energy, is that also a big issue for sperm whales?
Development of renewable energy. You mean wind farms? So wind farms, it's increasing vessel traffic, so risk of ship strike, and also mostly anthropogenic noise. And vessel traffic noise is also in the same hearing frequency of sperm whales, where they are affected by.
Okay. So this is also going to create an extra conflict, I guess, in this region too. Okay. Okay, I think I had one more. At the beginning you mentioned, I didn't catch whether you said microplastic or macroplastic, and I know we were just macroplastic.
Macroplastic.
Then it makes more sense to me.
Yeah.
You were talking about it affecting the digestive tract, and I didn't
Yeah. There's been studies about microplastic on sperm whales, but like the effects are not really understood, or I don't know if there's actually a lot of effect on the endocrine system or anything. Is not really clear. Whether macroplastic, we know that it can stuck the gastrointestinal tract and just result into death and stranding. So.
That's it for me. Thank you very much.
Thank you, Daisy.
Okay.
No, I think so.
I don't mind.
Go for it, go.
Okay, Marion, thank you very much for the presentation. Don't worry for the stuff, because we are all melting here and suffering. So you are doing great. Once again, congratulations. And yes, I was involved in one project with this sperm whale, and I was surprised how the biggest odontotest has such a little information, and particularly in the North Atlantic, that is one of the most studied regions. So I wanted to ask you if you really have the opportunity to continue researching in this area, apart from doing trips for spotting them. What else would you do to support your hypothesis? Like, this is a very important area for them.
Apart from presence detection, you mean? I think so. There's research in the Norwegian Sea about the social gathering that happens in certain moments of the year, mostly in winter. So we couldn't really see it from our expedition that were both of them in the summer. But I think it would be really interesting to actually go there in the winter, and especially with the passive acoustic monitoring, record probably the social vocalization, like slow click that they can do sometimes, or coders that have been recorded a few times as well, but not really described so often. I think there's still a lot to know about how they feed in this area. There's gonna be some more in this area with the team. I've been doing this study,
but we are not only focusing on sperm whales, we're focusing on noise-sensitive cetacean. So yeah, I just took this sperm whales part from the survey, but there's a lot also about northern bottlenose whales that are known to be even more sensitive than sperm whales to sound. And near the Jan Mayen, there's lots of them that I could see last week. Yeah, there are lots of them. So yeah, it's a really critical area to put more resolution there.
Yeah, you just got the direction to my next question, because I wanted to ask you, okay, you were there, you were spotting strong whale, but I'm pretty sure that there are many more other cetaceans and many other whales in that area. So normally we are used to see you want to protect some area, or you want to stop something, we will say like that. You base this into a ecological or these different species. So do you see other species? And but you already almost answered that question.
Yeah. So we saw most of the sighting and acoustic recording were northern bottlenose whales, which are the critical species talking about sound pollution. We have a lot also of killer whales there around that can be sometimes sensitive to noise as well. And also this area is known to be a really critical migration corridor for baleen whales, like minke whales that we cite, and fin whales as well. So yeah, it's not only toothed whales. Baleen whales can also be impacted, and as I said, deep sea mining is not only about noise pollution, but is also going to result in sediment plumes, a lot of impacts on the biochemical environment, and it's going to affect even non-sensitive, non-sound sensitive species as well.
And my last question, I think you also almost answered with the previous answer. And is if you know the prey, what they are eating in that area, because you mentioned that it could affect also the diet.
So they're mostly eating cephalopod up there and Gonatus genus, the genus like Gonatus, which is the cephalopod species. We don't know much about the impact of deep sea mining on this prey species. We do know that they could be impacted by climate change. Yeah.
Thank you very much.
Thank you. Okay, so first of all, I would like to start to congratulate you. I think you did a great job in the presentation. And I mean, I like many of your slides were self-explanatory, so congrats to that. I mean, it's really important when you achieve these kind of things, so well done. I have just a few comments or a few questions about that. About your review, I think many reviews is really time consuming and it's really important in some cases. You identify clearly the knowledge, knowledge gap, yeah, and that's great. And which keyword did you use? I didn't have the chance to read your manuscript, so this is the first time I'm seeing your work. So which keyword did you use for your review?
Well, there's a lot.
Because that's something that normally when I'm doing a review or some people working with me doing a review, it's clear to identify, and that I'm sure it's included in the manuscript, but you should clearly say. And instead of doing maybe review, what I would do is like more like a meta-analysis, because it would be nice to see, I'm sure you include in your manuscript maybe, it's like, okay, so out of all the different approaches that you identify, which of them, which is the different weight of data? Because you have 394, or correct me if I'm wrong, 394 papers that you review about. So, and you are mentioning different claims, so how well supported are those claims depending, I'm sure that not all of them are equally supported, or equally weight. So, I would try to do some statistics, even when you're doing some review, what you can do is like have like this typical, image of keywords and see how the relevant, the relative proportion of different terms that appears in the paper. So I will add some more statistics to that, even though it's a very, maybe you did.
Yeah, we were thinking about that when I wanted to do this review on male sperm whale, but the researcher I talked to about male sperm whale ecology were not certain there were enough papers in general to conduct more systematic review and meta-analysis. Yeah, so I didn't plan to do something that deep into details because I feared that I couldn't have enough data for that. Yeah, I mean 290 seems a lot, but it's really spread between all those sections as well that are quite clearly separated. And I also didn't include all of the findings of knowledge in this presentation because it would have been a bit too long, I think. Yeah. So.
Just the findings. So it's more like reflection, or which was your major challenge in your thesis, and the general question: what would you do differently if you have to do it again?
Sure. That's a good question. Well, if I had to do it again, I think in the perfect world, I would have liked to have more data, because this case study has been a very short time on field and a very few data, even though it's a lot of encounters for the time we've been there, and have a bit more powerful study, I guess, on the presence of spermers, and also include more aspects on that. Maybe study more their feeding behavior there and the social behavior, and have something a bit more holistic on also those different aspects, and also a different time of the years.
Okay, that's all. Thank you very much. Well done.
Okay, Mario, we are out of time. So you are done. Congratulations. Enjoy the night.
Thank you.